USA. He has graduated in the field of biochemistry in PSGCARE, followed by Master in Biotechnology in MKU Madurai. He was conferred with PhD in the molecular biology and works as a research fellow in the Institute of Molecular and Cell Biology, National University, Singapore. He moved to the Johns Hopkins University School of Medicine in the United States for the postdoctoral fellow in the Department of Neuroscience. He was bestowed with fellowship since his post-graduation and secured MSc Fellowship, Department of Biotechnology, India, PhD Fellowship, Council of Science and Industrial Research, India, Research Fellowship, Whitehall Foundation, Postdoctoral Fellowship in USA. Then he secured Andrew Virginia Faculty Fellowship, Washington University School of Medicine Award. Then he secured Swarna Jayanti Fellowship, Department of Science and Technology, India. He authored more than 30 journal papers and published in the high-indexed open access journals. He is a principal investigator for more than a dozen major projects and secured funding from governmental and non-governmental agencies. He prepares the teaching modules on neuronal cell biology and neural development and supervises the thesis for undergraduate, postgraduate, and research scholars in the United States and India. He chaired as a member in various committees of the institution. He has been invited as a resource person across India and other countries all through his career. He is one of the grant reviewer in DBT Task Force, BARAC, and appointed as a PhD thesis reviewer. He is one of the panel member in Journal Peer Review Committee in the distinguished journals such as Journal of Neuroinflammation, PLOS Biology, Nature Scientific Reports, Cell, Death and Disease, PLOS1, BMC, Neuroscience, BMC Biology, Cellular and Molecular Life Sciences, Frontiers in Molecular Neuroscience, and etc., etc., etc. So, best wishes to you, sir. So, we are awaiting for your provoking and mind-blowing talk, which is just after the lunch.
Okay, thank you for that kind introduction. And it's always the two slots that speakers hate to present is the one before lunch and the one after. Because you're too hungry when it's closer to lunch that you don't want to concentrate. And after lunch, you know what happens. Your food coma kicks in. So what's the most important organ in the body? Sorry? Brain? Good, good. If you had said heart, kidneys, lungs, I would have said yes. All organs are important, but you need one to rule them all, right? That's the Hollywood dialogue, right? One stone to rule them all. So that's what it is. You need one organ that rules everything, and that's the brain. So the work that we try to do is to try to understand how you can protect the brain from injuries and from neurodegenerative disorders like Alzheimer's and Parkinson's and ALS, motor neuron disease. So what's interesting is the size of the brain compared to the whole body. If you take the human brain, it's just about 1.3 kilograms for an average. But imagine what else and what all the brain can do. It makes you what you are. The individuality that you each possess comes from what the brain is made of. So if you look at the anatomy of a human brain, you have regions that have been specialized to do a whole variety of functions. You can have the ones for very higher mental function or higher cerebral functions, which is your learning, memory, your consciousness, and to know who you are. But you also have regions involved in sensory functions. The sensory part of the brain, the motor part of the brain, the sensory part of the brain tells you what you're feeling, the feeling of pain, the feeling of cold sensation. All the information goes to the brain. And then you act accordingly, and that comes from the motor part of the brain. So you have parts that are involved in physical sensation of your environment, and you also have emotional functions of fight or flight, knowing how to get away from threat. and normal visual functions, your audition, your ability to taste things, your ability to smell things, your ability to process all of these visual information, everything is governed by the brain. So it's important, that's why the brain itself has to be protected, and that's why you have a very dedicated structure called the skull, because it needs to protect this very, very soft, jelly-like organ compared to every other organ system that you see. And the problem that we face in countries like India is the aging population is really on the rise. So number of people aging is associated with a lot of increase in aging-associated disorders. We call these as neurodegenerative disorders. And primarily, these are caused by changes in the neuronal population where you start to lose neurons in parts of the brain that slowly become important for a lot of functions. One thing that we know of very commonly called the Alzheimer's disease or the memory disorder, it's not dementia. Alzheimer's disease is the most largest form of dementia in humans, caused by loss of very specific population of cells in the brain that is involved in learning and memory. And that's why people, when you age and you actively have Alzheimer's, you start to forget things, and you may start to forget smaller things initially, where you kept your keys, where you kept your phone. Then you start forgetting how you got out of the house and how to get back to your house. Things that you take for granted on a day-to-day basis, you start losing, and the worst part is what happens towards the end state of the disease. You forget who your wife and kids are. So you can't even remember them. And that happens by very specific changes that goes on in the brain. And the next common one that we see in India is Parkinson's, and in the world. This is the second most common neurodegenerative disorder. Happens again by specific loss of a particular population of cells that controls motor functions. And this disorder is called a shaky palsy, where you suddenly start to get these shivering, and with time those neurons will die, that you won't be able to even pick up a glass of water to drink or a spoon of food to eat. And slowly what happens is your normal motor functions is what is affected. And the last of the motor functions to surrender to this disease is your breathing. When you're concentrating on something else, you're not concentrating on breathing. It just happens naturally. But there are stages where you have to manually, or rather coordinate your breathing. When you lose control over that, that's exactly what happens. Swallowing is also a voluntary control. So those patients slowly die because of loss of these neurons that control motor functions. Then you have Huntington disease. Again, cell death in a very particular region also controls motor function, different from Parkinson's. But you see in the electrical wires, the protective covering around the wire, as you see in the brain, will start to loosen, come off by various factors, and the neurons will not be able to function properly, and then those cells will start to die, and that's one of the causative agents for. And of course we have the ALS, which is also called as the motor neuron disease. We have motor neurons not just in the brain but also in your spinal cord. These are the ones that controls your peripheral movement. And these neurons, very specifically, all these disorders have a genetic component to it. And one thing that has been baffling to neurobiologists is when the whole ALS became pretty much a poster child for people to know. Your Stephen Hawking, the most celebrated astrophysicist, suffered from a form of ALS. He was a luckier one because he was having a form of ALS that could still give him life to live, even though the of life, as we know, is stuck to his wheelchair for the rest of his life. But not everybody is so fortunate, because ALS is almost like a death sentence to you. When it hits you early, you got about 5 to 10 years to live, because same thing, motor functions will start to shut. Eventually you won't be able to swallow, you won't be able to breathe. In fact, you choke yourself to death, usually in sleep. So the problem here is all of these are in the rise in developing nations and also developed nations with the increase in the aging population. Number of people getting old is higher, and exposure to a lot of carcinogens, etc., could actually lead to any and all of these disorders. And also in comparison to that, you have these physical trauma or injury to the brain, what we call as stroke, which is caused by very many factors. Cardiovascular is the major driver that leads to where your arteries get clogged in the brain, and suddenly either you have what's called as an ischemic stroke, blood clot, no blood supply, cells die, and neurons in that region starts to die. Or you can have a hemorrhagic stroke, where because of high blood pressure, a very small artery or can actually burst, spilling blood in the brain. That pressure will also kill these neurons. Or you have your what called a traumatic brain injury caused by concussion, falling off a bike, being involved in an accident. All of those will cause these changes, damage physically to the brain that would arrest a lot of important functions, paralysis and whatnot. So the question is, how do we prevent these neurons in all of these conditions from dying? And before we get to that, we need to know what is it that this brain is made of? What are all the components of the brain? So there are many cell types in the brain, about a dozen cell types, but the most important ones that we speak about often is because of their sheer numbers is your neuron, which is the most important. Everybody, when you say nervous system, you think of neuron. That's the major cell that helps in conducting information. And then you have your astrocytes, and these are called astrocytes because they look star-like in appearance when you stain them and look in the brain. And they do a lot of functions, about maybe 10, 12, 15 different functions. These are the most abundant cells in the brain in terms of sheer number. You have oligodendrocytes. I said these are the insulating cells that you see, just like your plastic around your electrical wire. So they insulate the neuron so that your conductance is very fast. And when this covering that you see in blue is lost, that's when you get multiple sclerosis. And another cell type called the microglia, tiny cells. These are the immune cells in the brain. The brain has its own surveillance systems, the immune system. So the microglia is not produced in the brain. All the other cells are produced within the brain. The microglia is formed in the yolk sac during development. And at early ages of development of the embryo, these microglial cells come through the bloodstream, get into the brain, and then establish themselves. So they are more like not citizens, but they're more like permanent residents of the brain rather than being born citizens of the brain. Among these, I said the most important cells, in my opinion, and because not just because we study, is because of their versatility of function, are these astrocytes. And one particular response of these astrocytes that is very pertinent to all the disorders that we saw or the injuries that we went about is what's called as a astrogliosis or reactive astrocytes. So these are like your immune cells. When you're in your periphery, when your body gets a bacterial or a fungal or a viral infection, your macrophages and your T cells will get reactive. They become activated and they start doing all they have to do, producing anti-inflammatory molecules. They also have inflammatory molecules. You're going to have antibodies being produced. Very similarly, when the brain undergoes change, either physically in terms of trauma or neurodegeneration that comes in, these cells get triggered by a whole lot of growth factors to become a form that we call as reactive astrocytes. They become much larger in size, the one you see on the right. They become much larger and they start producing things, with the whole idea being that we need to protect the brain from undergoing damage and death. Typically, it's a helpful process, but what happens is when the disease extends beyond a particular time frame, these cells themselves may start to contribute to the disease because their normal function starts to break down. So the whole idea is what regulates these astrocytes to go from left resting state to the right active state. So what are the functions of these reactive astrocytes? These are a lot of neuroprotective functions that they do, is to clear debris. And if it is infection, they try to clear that infection. If it is a bacterial, fungal, or a viral infection in your brain, they try to clear that. They repair the blood-brain barrier. The brain is a very, very secluded, protected structure in the organ in the brain. There is actually a barrier called the blood-brain barrier. The barrier is extremely specific Not anything can get into the brain as they wish. To give you an example, even water, for it to go into the brain, has to go through a channel. So water just can't go into the brain. There are channels called aquaporins, which actually transfer water from your blood into the brain. The glial scar, when you have a big trauma, they form the scar tissue like you have a scar forming on your skin after an injury, and they prevent infiltration of peripheral cells into the brain. So they help in reducing inflammation. Inflammation is good initially, but it becomes bad with time, so these cells help in regulating both. And toxicity of glutamate. Glutamate, the amino acid glutamic acid, is your major neurotransmitter. 90% of your brain cells, neurons, use glutamate as a transmitter. And that glutamate, usually when it gets secreted by one cell to activate the next neuron, that's when you have conductance happening. But this glutamate has to be removed as fast as possible. If not, you will end up with a condition called epilepsy. If the glutamate is going to stay in the synapse for long, it's going to keep activating your next cell, which is called the postsynaptic cell. And that's one of the reasons you have epileptic seizures, because the cells get activated constantly, leading into these epileptic episodes. So they help in actually removing excess glutamate. So they have very specific transporters for glutamate, and they stop these transporters. So we know a lot. I'm going to cut fast through all of this. We know a lot about the signaling molecules involved in this transition from sort of a resting state to this reactive state. I will cut through this faster. And what we know, initially I said these cells become reactive in all of these conditions, and we all thought for all these years, about 50, 100 years before it was discovered, that they all respond in a very similar manner. But with the advent of better technologies like the RNA sequencing, they started looking at whether these changes are the same or are they different. So a group in 2012, they took the brain and challenged it with a bacterial infection called lipopolysaccharide. These are the cell wall of bacteria that stimulates your immune system. So you just inject it into the mouse. The astrocyte senses these particles of the cell wall and think that there is a bacterial infection, and they become reactive. On the other hand, they gave a stroke. So it's very simple. You have this carotid artery that's the major artery on both sides that supply blood to the brain. So we have a means where we can put a small wire and cut out the blood flow to parts of the brain, mimicking stroke. When you do that, cells die, of course, but then it also activates these cells, astrocytes, in response to those cells dying. So when they took these two cells, in the red and green, both astrocytes, looked at their gene expression pattern, they found that the expression pattern was very different, indicating that they may show very similar appearance from outside, but when you look into what they are expressing, changes in their gene expression, it's very different. So one they found to be neuroprotective, the one in green caused by ischemic stroke. The one caused by bacterial infection was found to be neurotoxic because they were showing signs of secreting something that kills neurons as well. So work subsequently showed if you block the red cells kind of astrocytes from forming, this was actually neuroprotective. So a lot of papers came about in the last 5 to 10 years that show that if I block this transition to that red kind of neurotoxic cells, the brain is better protected in Alzheimer's and Parkinson's and in stroke models. But what are the regulation that makes them good, makes them more protective, was not very well known. So the question we started asking in my lab is, what are the mechanisms by which you can make these astrocytes become protective? And whether if you can make them protective, can they be broadly protective, or they're going to be very narrowly protective only for certain disease or injury but not for the others? That was the question. So we started off looking at this transcription factor. As you all know in this audience, transcription factor is something that goes on to activate transcription of a very specific set of genes. And we started working in this for quite a while before that to look at how this transcription factor regulates brain development using mouse as a model system. So everything that we do in my lab uses mouse as a model system, transgenic mouse and knockout. So this is a transcription factor that binds DNA on very specific promoter elements and then activates downstream target genes. And by using complex genetics, we can delete this transcription factor in very specific cells in your body. If you delete SRF, the serum response factor, in every cell in your body, in the mouse. The mouse dies around embryonic day 7 or 8. It's about missed gestation. So it lives only for about a week or so, and then the pup gets aborted. So it cannot live. So you cannot remove this gene from every cell in the body. So we do molecular genetics where we can cross these. One mouse will have a means of deleting this. The second mouse will provide you with the key to knock it out in a very specific manner. So when you do that, you end up getting with a mouse wherein we can delete this gene only in the stem cells in the brain during development and nowhere else. And that's exactly what my first grad student, or rather second grad student, Paul, did. So, and then when we did this, I'll just cut the story short. What we found was the astrocyte numbers, the major cell type I'm talking about, numbers dropped by 50% when we delete this transcription factor, indicating that this transcription factor is required to make stem cells become astrocytes in the brain. But unfortunately, this mouse dies within 18 to 24 hours because of cerebral hemorrhage. It shows a lot of blood flowing within the brain, and the mouse almost dies by 18 hours from birth. So what we did was we used a different approach. We took another enzyme, recombinase, coming from a promoter that is only expressed in astrocytes. So now we deleted this gene only in the astrocytes in the brain during development. Okay? So when we did this, now we get a mouse that doesn't die, thankfully. And then we now asked, what happens if I remove this gene only in the astrocytes? And we stained them for different markers. GFAP is a particular protein expressed only in these astrocytes. ALD is also a protein that's expressed only in astrocytes. When we do that, this is the control brain. We looked at 1 month of age in a particular region called the hippocampus. That's the structured in red box that is involved in learning and memory, both in rodents and also in humans. So the hippocampus is a major structure for learning and memory for all of us. Your spatial learning comes from this particular structure. When we looked at the mutant brain, what we see is this increased expression of this protein called GFAP, and also increased expression of this protein called ALDH1L1. It indicated to us, based on literature and other studies, that the astrocytes are becoming reactive, becoming larger. Mind you, this brain is not challenged with anything. There is no disease, there's no damage. When we just remove this gene, the cells start to become reactive. And we find this followed by my other grad student, Monica, who characterized this mouse. So you can see this phenotype is shown in every cell in the every region in the brain. It's not restricted to the hippocampus. Wherever we look at the neocortex, the hippocampus, striatum regulates your motor control. Corpus callosum is the structure that connects both your brains, both halves of your brains. So every part we see these astrocytes are becoming reactive, and they stay in that state throughout the life of the animal. So it's like removing the brakes and making you constantly angry. Nothing to trigger you to become angry, but I've just removed something that you're in a perpetual state of anger, right? Change of shape, change of function. And we confirmed this with other protein markers. And the next thing we wanted to, again, confirm that they are indeed reactive. We did quantitative RT-PCR for certain genes that have been shown by other papers. So all these pan-reactive genes were up, and the bacterial-induced genes are also up. The stroke-induced genes are also up, indicating that our astrocytes do not fall into the infection type of astrocytes or the stroke kind of astrocytes. Then what are they? Then we wanted to make sure they're not harming the brain in any ways. So we have done a lot of experiments that showed that the neurons are not dying. So neurons are still there. So the astrocytes, even though they may look rogue, they are not doing anything that affects normal neuronal structure. The next we looked at is blood-brain barrier. The astrocytes are extremely important for maintaining this barrier I spoke about, where even water has to come in. Not every molecule you take can go in. Even for glucose, it has to be transported into the brain. So the way we test this is very simple. What we do is we open up the heart when the animal is anesthetized. We inject a very small dye into the left ventricle for it to circulate. It's a dextran 10 kilodalton fluorescein. It's a fluorescent molecule. Fluoresces green in color when you shine UV light. And what we do is we let it circulate for 5 minutes, and then we sacrifice the mouse, take the brain, and we ask how is the blood-brain barrier doing. Because like I said, even though it's 10 kilodalton, it cannot pass the blood-brain barrier. So if your barrier is very good, you will only see the staining within the blood vessels and not into the regions of the brain. And that's exactly what we saw. Everything in the control and in the knockout is within the blood vessels alone. All the green structures you see are your blood vessels in the brain. As a control, we do what's called as a stab wound. We open up the brain, put a needle, take it out, and then you now break the barrier, and you now start to see the green part leaking. The line that you should see is the way the needle actually went in. So we did another dry called horseradish peroxidase, which is 44 kilodalton. Again, we don't see any leakage into the brain, telling us that the blood-brain barrier is not affected by these abnormal state of astrocytes. Then we looked at synapse numbers. Synapses is where two neurons communicate with each other. Long story, synapse numbers don't change. And in light with that, electrophysiology in terms of how these neurons communicate doesn't get affected in the brains of these mice. This is all important for us to make sure that the astrocytes that are in a very abnormal state are still able to provide normal functions, and they themselves are not being harmful to the brain. Then we looked at behavior in these mice, and we test open field is basically we have this large box where you let the mouse roam around and you see whether there's an abnormal way in which they move around, and we don't see anything abnormal between these two. The next is a contextual fear conditioning. The way we do this is we have its home cage, and then we have a cage in which we provide cues in the cage. There will be one or two signs put in the cage, and it has a metal floor. The mouse goes in and explores. At some point it gets electric shock, a very mild shock, but enough for it to feel the shock from the surface. It's like everybody. If you get beaten by your teacher, the next time you go to your classroom, you know this is where I got my beating. So the mouse also remembers the next time you don't have to give it a shock. You train this. This is what's called as a Pavlovian conditioning. And as conditioning, fear conditioning among species, all you need is one time. You don't need to be trained over and over again for a fear condition. One time you'll remember for the rest of your life, right? So that's what we do. So we give a mouse a training context, we put it into another context, give it a shock, the mouse remembers that cage. The next time we put the mouse, the next day, 24 hours later, we just put the mouse no shock. The moment it sees the cues, the mouse freezes. All this is real-time recording happening, and we call what's called a freezing response. The faster it freezes, the faster it remembers. The slower it freezes or doesn't freeze at all tells you that it has forgotten what it learned a day before, just like us. So what we found is there was no difference in this freezing response between the control mouse and our mutant mice, indicating that this memory is not affected in line with what we saw with synapse numbers being same, synaptic transmission being same. Another memory, this is again spatial memory, a second type. What we do is we have this wheel with holes around it, all the black ones you see, these are called a Barnes maze. Only one hole has an opening to get into a box. The rest are all closed holes. And we provide cues around the room, and then we train the mouse, and after about 3-4 trials over 4 or 5 days, this is not like the fear conditioning, this one needs to be trained for about 4 or 5 days. And then by looking at the cues, it knows exactly which hole to go to and get into the box underneath, because they hate bright light and they like to stay away from the bright light. So we trained that, and what we found is both the controls and the mutants especially was able to find the escape hole in the same time without any delay, indicating the spatial memory is intact in our mutant mice. So all of this is good. Then we asked, in aging, one interesting thing with the brain is as you age, there are certain structures in the brain that are in itself vulnerable to changes. And one particular change that happens with aging is loss of neurons. The neurons just die, and one structure is your cerebellum, this part that's right at the back of your head before your spinal cord starts, and that controls your motor functions. And what you see here in green, these dots are particular cells called the Purkinje cells, and these start dying in a mouse by about 12 months, 1 year. The mouse can live up to 3 years. So at 1 year, these cells will just start to die by itself. And in humans, you also have this condition. Human also starts to lose. That's why your motor coordination, motor control starts to weaken as you age, because these cells naturally die. But when you look at our mutant mice on the right, we see more of these cells present, indicating that these cells are these astrocytes are able to arrest death of these Purkinje cells. How do you measure that? And we measure that by doing what's called as a rotor rod test. This is a rod that will be rotating at a constant speed First we trained the mouse, you put the mouse, it's going to be managing to balance itself as the wheel is turning. So we trained this mouse on 3 consecutive days where the speed of the rod is at about 5 rotations or 5 RPM per minute, 10 RPM, 10, 20, 30 RPM we do. And on the test day, what we do is we increase, it goes gradually increasing instead of a fixed speed. Starts off at 5 RPM, then goes to 10, 15, 20, 25, and we see how long does the mouse stay on these rods without falling. And the moment it falls, there's a sensor at the bottom here, the 2 mice fallen, and the clock automatically stops. The moment it hits the bottom, the clock starts for that mouse. So the longer a mouse stays, better motor coordination. The faster it falls, poor motor coordination. And when we do that, we see that our mutant mice, which had more of those green cells, are able to hang on to that rotor rod longer, indicating that they're able to have better motor coordination because of more cells still alive and not dying. So that's an indication that even aging, we have a lot more data. So aging associated decline, changes in the brain is also better protected in our mutant mice. And finally, we also wanted to know, because one thing associated with aging is also memory loss, memory decline, not Alzheimer's, normal dementia, right? So we wanted to make sure whether that is changing in our mutant mice. So we did the same Barnes maze, and then what we see, long story short, is when you train them for 4 or 5 days, test them after a day after the last training, they remember better. Both of them remember the same, the controller knockouts. What we do is give a month gap and come back and ask, are you able to remember what you learned a month ago? And when we do that, we found that the mutant mice in red is able to remember the whole better than the control does. The control is already beginning to forget where the escape hole is, whereas our mutant mice were able to remember where the escape hole is present even after 1 month of training. Nothing in between. You train, you test, go sit. It's like giving you a retest for your lessons a month later. How much do you remember? These mutants remember better. And finally, we've done gene expression studies to see what is changing in these astrocytes, and we found genes that are involved in defense response against bacteria, against virus, and interferon responses, which are normally said to be neuroprotective in nature. So last few slides, most exciting ones. So I presented you with all of these changes the brain can undergo, damage to disease. The main question for us is, are these astrocytes able to protect the brain from these changes? So for brain injury, we took traumatic brain injury, a neurotoxic model, and then a stroke model. And for neurodegeneration, we took Alzheimer's and Parkinson's. For traumatic brain injury, this was done by my postdoc, Surya. The way we do this is we anesthetize the mouse, put it into a stereo taxi that gives us coordinates, just like a GPS. We know where exactly to go and inject something that we want to inject within the brain. And then we put a needle inside, take it out. Don't worry, your brain doesn't feel pain. So the most interesting thing about the brain is you have pain receptors everywhere in the body, but not in the brain. You can stab the brain as much as you want, you will not feel pain. The pain comes from cutting the skull open and piercing the skin through. But as such, the brain itself doesn't have any pain receptors. The mice they are always under anesthesia, they are under very heavy painkillers, so they don't feel any pain. So when we do this, and that's how we expose it, the dot that you see is where the needle was, so we have to shave the skull a little bit so that we can poke through. And we do that, we then look at how the wound is healing. So the way the wound heals is from inside out. So you got a whole needle went in, and the brain starts to heal from closest this way. So when we look at how fast it's healing, you have to see how big is your cavity. If the cavity is bigger at a given time compared to the other, then one is healing slower than the other. So that's exactly what we measure. And this is a top-down angle. And you can see here that the mutant mice have a smaller hole, indicating that the wound is healing faster in the mutant mice compared to the control mice. The next is we used a toxin that actually produces seizures in the brain called canic acid, very similar to glutamate. And this is why for some people they are very sensitive to a chemical called MSG, monosodium glutamate. That's commonly used in Chinese cooking. And some people are very sensitive to it because it is phenocopies in terms of function your normal glutamate and can cause seizures. So that's what we do. We actually inject it right into the brain, and once the mouse comes out of anesthesia, it undergoes seizures, and we score the different kind of seizure that it shows, and typically the mouse recovers in 1 hour because it wears off, the brain clears it, so the mouse. But what happens is, this is a neurotoxin, so it starts to kill cells in a very specific part of the brain called the hippocampus, involved in your learning and memory. And we look for cell death. What you see on the top panel is your control brain. All the green structures you see are dying cells and dying fibers. What you see in the bottom is what we get from our mutant mice. We get almost no cell death in terms of when you induce cell death by canic acid. We looked at another way of looking at cell death, which is looking directly at broken DNA within the cell, because when the cell dies, undergoes apoptosis, so the DNA starts to break down, and you can stain for that broken DNA by labeling that DNA, called end labeling. And all the green dots you see are the DNA that's broken down in the cells, indicating that those cells are dying or dead. In the mutants, we actually see very, very less cells that are dead or dying, indicating that these astrocytes are able to protect the brain from epilepsy-like condition. Then we induce stroke. We use a different stroke model. What we do is we inject a dye called rose Bengal dye. And the dye has a very specific quality. When you shine a light of a particular, green light of a particular wavelength, about 590 nanometers, it oxidizes, causes platelets to aggregate, causes stroke. Blood clot. So what we do is we expose the brain. We can actually see the arteries. We inject the dye 15 minutes earlier. We shine a very tiny 1 mm or less of the green light in that structure for about 15 minutes, causing blood clots to form, and the mice undergoes a stroke. When you do that, this is a control where we take the brain, section it, and stain it for a dye. And you see all that white patches you see is where your stroke infarct is, where all the cells, the neurons have died. So that tells you the extent of cell death, where we had blocked the artery. When you look at the brain, those white spots are still there, but they are smaller and in fewer brain sections, indicating that our astrocytes are able to protect these neurons from stroke-induced death. So the question is, this is all good. What about neurodegenerative disorders? Can we actually help the brain from neurodegeneration? And the first thing we did was a model for Parkinson's disease. So Parkinson's disease, the cells that die is in the very last 2-3 slides. The cells that die is in a region called SNPC, the substantia nigra pars compacta. These are the very small population of cells that specifically die in Parkinson's disease. These neurons send their projections to the front part of the brain called the striatum, which controls motor control. Motor movement is controlled by the striatum. So when these neurons die, they don't send their projections to the striatum, so motor control is lost because they secrete a neurotransmitter that activates the striatal neurons. And those neurons don't die, but they are not able to be activated, and hence you have the typical Parkinson's symptoms of shakiness and loss of motor control with time. So we inject a dye, a neurotoxin called 6-hydroxydopamine, which is an analog of dopamine, the neurotransmitter used by this. And after a week of 10 days of injection, we do it only on one side. We don't do it on both sides. If we do it on both sides, the mouse dies because it has a very, very drastic motor phenotype, so it won't be even able to eat or move around. So we only do it on one side. So it gives you a partial paralysis on the other side, right? Just like you have stroke on one side. When you do that, then we stain for these neurons. You can actually see the cells are lost with time in that 10 days. When you look at the knockouts, we see almost no or negligible cell death in the mutant mice, and that's quantified here. So in the mutant mice, injecting this neurotoxin doesn't cause any death of these dopaminergic neurons in 6-hydroxydopamine induced ones, but it kills neurons in the control mice. That is good. Do we have a phenotype for it? So we changed our strategy. We injected into a striatum, and we do what's called as a rotation test. So we are injecting only on one side, I told you. So the neurons in that side will die. So that part of the brain will not get any inputs. Now you inject this mouse with a drug called apomorphine. It's very similar to morphine, and it actually activates cells on the side that you have deleted, which is the right side. So what happens is the side in which you had killed the neurons, the next target, which is the striatal neurons, get very active when you give apomorphine, and the mouse will start to turn on the opposite side. So it's a very simple experiment. You put it in a beaker, inject this dye, within a few minutes, the mouse will just start to keep going round and round and round towards the opposite side of the lesion. So what we measure is how many times it turns, does this productionum every 10 minutes. And we compute it. And the lesser you produce, lesser cells dying. The more you have it, more cells are dead. That's exactly how we measure behavior. And when we do that, we see all the blue ones are the control. So average number of rotations per minute, you get 20, 30, 40 rotations. With time, we do it every week after we kill these neurons. And in the mutants that you see, you hardly see the orange ones. They hardly go in circles, indicating that these neurons are not dying in our mutant mice, whereas the control neurons are dead, and therefore they show this rotation phenotype. The second experiment that we do is what's called as a cylinder test, because like I said, it's a stroke. When you kill one side, you cannot use the other arm to reach out, because this is paralyzed. So what we do, if you put a mouse in a beaker, it will try to reach out with both limbs. So we count the number of times it's reaching up with its right and left, and not using one side. So what you see here in orange is the side which won't be able to use if the cells have died on the other side that you have injected. So when we did that, what we see is our mutant mice is able to use the contralateral, the other side of the limb, better than the control mice dies, indicating again behaviorally that this cells not dying is beneficial behaviorally in terms of both the apomorphine test and this. They are less paralyzed compared to the control mice. Lastly, we looked at, this is just to show cells not dying in control versus the mutants that matches with the phenotype. Finally, we crossed our mice with a model for Alzheimer's. One of the major hallmark of Alzheimer's brain is the formation of these plaques called as beta amyloid plaques in the brain that just aggregates outside the neurons, and with time this just increases, starts to kill neurons, starts to impede with neuronal functions. What you see on the top stain for this is that the plaque numbers are highly reduced in the mutants that you see in the third and the fourth column are the mutants, and you can actually see the plaques are lesser in size and also fewer in numbers, indicating that these astrocytes in the brain are able to clear or help in the clearance of these plaques much more efficiently. So our mutant astrocytes, as far as we can test, is able to protect the brain better from physical damage, stroke, injury, or also help in protecting the brain in neurodegenerative disorders, Parkinson's and Alzheimer's. So to summarize, so we are able to delete a particular transcription factor in these cells called astrocytes that produces a phenotype that becomes reactive, and these reactive astrocytes are not harmful to the brain as far as we can tell. However, they seem to be very broadly neuroprotective. So we are expanding this to look at motor neuron and Huntington, etc. The problem is getting access to these mutant mice to doing those experiments, but that's sort of where we are. So now we have a very good target for gene therapy approaches. We cannot inhibit SRF because it's expressed in every cell in the body. So you cannot pop a pill that's going to inhibit this protein. So we are developing viruses that would deliver a CRISPR RNA or a dominant negative protein, so that we could, and in a very astrocyte specific manner, that we could either inhibit this protein or knock it out completely, so that we can now make it reactive and hopefully see benefits in human patients as well. So the idea next is to move towards human astrocytes, human iPSC derived astrocytes, and start doing transplantation studies. We delete this protein in the human astrocytes, transplant those cells into the mouse brain, and ask whether if we do all of these conditions, whether the human astrocytes deleted for this protein is also neuroprotective. Well, that's the first step for us to convince ourselves we have something that could be potentially useful for gene therapy. If that works, the next step is to take viral vectors and start deleting this in a human condition. We can either do it localized for Parkinson's or a little bit more broader depending on the disease. Thank you very much. And to acknowledge, work was done by several grad students. Paul started this work, followed by Monica, and then postdoc Surya. Srividya was a technician who's now a graduate student at State University, New York, Buffalo. Sumita and Priya were again technicians that helped. And the other grad students that were on other projects, my collaborators, James, Vijaya, and JNC, and my old colleagues and collaborators, David Gutman and Susie Baker, and funding. None of this would have been possible without good funding from both IASC and the Department of Science and Technology, Department of Biotechnology, and also CIFIC grant from the French government. Thank you.
Thank you, sir, for enlightening the young minds with your research innovation and creating awareness on manifestation of neurodegenerative disease. And thank you for sharing your interesting experimentation on behavior aspects of mice.
Now, we request the chairperson, Dr. Kalpani Ratnayak, to outline this technical session 3.
So, good evening to all of you, and we were able to listen a very comprehensive talk on reactive astrocytes in neuronal injury, aging and neurodegenerative diseases by Dr. Narayan Raman, Associate Professor, Center for Neuroscience, IISc, Bangalore. So, he has nicely summarize about the brain anatomy and the functions, then the neurodegenerative diseases, type of strokes in the brain, and the neurons and the neuroglial cells, and also some animal studies he has done with SRF deficient reactive astrocyte. So thank you very much, sir, for your very comprehensive and very clear, informative talk. And now the participants, you can ask question from the resource person.
No, it's okay. It's easy to keep walking.
See, students and participants and delegates, we have such a big participation. It's our duty to ask the questions with such a comprehensive talk, and the persons of caliber from IASC, even though he is our own alumnus, of such a caliber, high caliber talk he has given. So kindly interact so that you also develop the ability of interacting with the resource persons. More questions we expect from the participants, PhD students, as well as the outside participants. Come on. Our own MSc students, all of you should interact with the speaker, definitely. Come on. Our second MSc.
Nothing is a simple or a stupid question, so don't hesitate.
You know, he is not a stranger. He's our own alumnus. He studied here. And
to ask, yeah. In fact, you guys are better than what we were 30, 40, 30, 35 years ago.
Yeah. In fact,
more open, you can voice your thoughts, clarify doubts.
Good afternoon, sir. Yeah. First of everything, thanks for a wonderful presentation.
Thanks.
Like you explained the things which are very clear manner. As a third UG student, I can easily understand what you did for the mice and how you approached in the mechanical injuries as well as the neurodegenerative diseases. Mm-hmm. My question is, unlike other cells, other glial cells, why you chose astrocytes? Could you explain it in a further way?
Yeah, so very good question. So I didn't have time to get into the functions. So for a long time, but until 10, 15 years ago, astrocytes were considered to be scavengers in the brain. Neurons were given more importance because when you think about anything that the brain does, you think of neuron as the first cell that does it. And oligodendrocytes are mostly considered to be just insulating cells. They just cover up, protect the brain from losing transmission. The astrocyte, if you look at the functions before, they provide nutrients, they remove excess ions from after the neurons have finished, clearing up all the debris. So they were considered to be basically your scavengers, things that are just around to be cleaning. And they're called glia because when it was stained about 100 plus years ago, they looked like cells that kept, glia means glue in Greek. So it looked like cells that sort of held the brain together like a glue. That's what they called as a glia. The astrocytes label came later. So their functions are only thought to be doing this, but in the last 15 years or so, people are able to now show that the astrocytes do far more than just mopping and cleaning. So they actively regulate a lot of important neuronal functions. Work, in fact, in the last 3-4 years have shown that you could activate astrocytes in a different way, not this kind of activation, but physiologically activating them. And you can improve memory in humans, in mice actually. And what gave them prominence was this interesting finding. So when people were analyzing Albert Einstein's brain, there's a part of the brain we call as a frontal cortex, called the medial frontal cortex, important for your intelligence. When they analyzed the cells in the brain slice of Albert Einstein, they found that this frontal cortex part had 3.5 times more astrocytes than neurons. So that sort of made people ask, Einstein having more astrocytes than to neurons in the major part of the brain involved in intelligence, does it mean that your intelligence is actually coming from astrocytes and not from neurons? So that piqued the interest, and then people started to show that it secretes factors that helps these neurons form connections. It secretes factors that actually modulates how the neurons transmit information. And now we also know that they can actually regulate in a multiple different ways. So these cells were very poorly studied, and what in the field we call as a neurocentric view. Anything in the brain is because of neurons. Right? So the people like me moving into the field were very less, also because not many things were known, so it's much easier to establish. Right? So that's exactly why we moved into astrocytes, only because now we are able to appreciate that even higher cognitive functions like learning and memory is very actively regulated by astrocytes, that you can now modulate their function to increase memory as well. So that's, and also these cells are very robust cells. The first ones to die are the neurons. Any perturbation to the brain, the neuron dies. When you have these stroke, etc., the astrocytes don't die. The astrocytes are very capable of anaerobic survival, which the brain cannot. Your brain cells need oxygen. That's why when you have a stroke, whatever it is, 3 minutes is all that they give you. Right? When you're drowning, etc., you got to be rescued. Your heart has to start pumping in about 3 to 4 minutes, after which you go into a state where you're brain dead because most of the cells will start to die. No oxygen, no glucose. The astrocytes can survive for much, much longer without oxygen and glucose. So that makes them very interesting, robust cells to do. So we are really pushing the boundaries to ask, what more can these cells do? So that was our interest, and that's why these astrocytes are more important, because they do about more than 10, 12 functions, very important ones, than just the scavenging, cleaning functions. Yeah.
Thank you, sir.
Good evening. Very wonderful talk, sir.
Thanks.
So I'm from clinical side. There's one question, might be it is out of syllabus, subject also. For any critical care patient, we will give glutamine. And the patient affected with single or multiple organ disorder, we will stop. How about this in any neurological disorder or traumatic brain injury?
Dying, and they will start dying, even in stroke. Right? By the time you know it's a stroke and you rush to the hospital, anything around the stroke area, the cells are dead. Those neurons are not going to come back no matter what. But what is more important is that the stroke cell death doesn't spread around to the healthier part of the brain. So the faster you can prevent that, you can save most of the neural functions. And the brain is extremely plastic. So a stroke patient, if can be given the proper physio in the next days to weeks, can actually regain 80-90% of whatever is lost in terms of motor control, etc. The brain learns, and there are parts of the brain that can actually take over these functions very, very beautifully. So in that instance, so what we hope we can do, because we don't know who's going to have a stroke, who's going to have a traumatic brain injury. So the question is, what we have is to just show that these cells can protect. So the next question we want to know is, if you induce stroke first and then make these cells become reactive, can they now reduce the extent of stroke and the extent of damage that is being done to the brain? That's the only scenario where we can think it would become useful for humans. That's there. So neurological disorders, which is a very interesting point, is now we are actually beginning to see astrocytes playing a very major role in cognitive disorders like depression, anxiety, post-traumatic stress disorder. Astrocytes have a very, very important role to play. In fact, when you take antidepressants, Valium, whatever, these astrocytes actually change their morphology and function. So one of my colleagues in the department actually studies how these astrocytes are changing when you are taking anxiolytic drugs for antidepression. And these affect these astrocytic functions. And people are beginning to show if you block some functions of astrocytes, you can make the mouse depressed. So that's coming up. Multi-organ, unless it feeds into the brain where the brain starts to sense, these guys can actually do that. So I said blood-brain barrier, and very similar question, and also the previous student that asked. There are parts of the brain where there is no blood-brain barrier, part of the brainstem, where these astrocytes process directly senses your blood. It actually senses the pH of your blood, senses the CO2 levels in your blood, and regulates your breathing. If the CO2 level is down, your breathing changes to CO2 levels are high, then it increases the breathing function. So they do a lot more. They help in circadian rhythm, your jet lag. Why you wake up at a given time every day and go to bed at a given time, you feel sleepy at the given time. So the astrocytes regulate circadian clock as well. Feeding behavior also, people are trying to see how much these astrocytes regulate feeding behavior. When you get hungry, when you feel full, etc. So they are very, very versatile cells. Yeah. I hope that I answered the question.
Yeah.
Okay.
Thank you, sir. Now, on the behalf of the organizing committee, we request Dr. Kalpani and Dr. Rajan, Associate Professor, Department of Biochemistry, to honor our speaker with a memento.
Thank you, sir. Thank you, ma'am. There's a saying that goes, be honorable yourself if you wish to associate with honorable people. We wholeheartedly thank the speaker, chairperson, and co-chairperson for making their way in technical session 3.
Now, we proceed for the next session of the day with a famous Thirukural saying on medical wisdom. Diagnose the disease and understand its seeds. Identify the cure and make sure it succeeds. With this, we humbly look up to our respectful resource person, Dr. Ponmariyappan Swami, scientist, DRDO Gwalior, to join this session as a speaker. With pleasure, we invite Dr. Naren Ramanan to please chair the session. We request Dr. Krishna Veni, Department of Microbiology, to join the session as co-chairperson.
On behalf of organizing committee, we welcome our speaker, Dr. Ponmariyappan Swami.
Good afternoon, everybody. It's a pleasure to introduce the speaker for the session. Dr. S. Ponmariyappan, Scientist, F and Head Project Director, DRD 208, is in the Biodetector Development Test and Evaluation Division of Defense Research and Development Establishment, Gwalior, India. His current research interests are in the development of biological detectors, biological testing facilities, and early warning systems for biological warfare agents, and also in the development of diagnostic kits for molecular immunological agent against botulinum toxins. He did his PhD in Manonmaniam Sundaranar University, Tamilnadu, guided 5 PhD students, and have more than 30 publications in the national and international peer-reviewed journals. Dr. Ponmariyappan has 10 patents filed and have received several honors, awards, and accolades. To mention a few, Dr. Ponmariyappan has received Laboratory Scientist of the Year Award in 2018, received Director's Award in 2017 for NBC protection measures during Independence Day celebrations 2017 at New Delhi, received the Director's Best Performance Award in 2016 for the development of early biological warning system, received the American Society of Microbiology Best Poster Award in the 52nd Annual Conference of AMI, Punjab University, Chandigarh in 2011, received the Brig KM Rao Award in the year 2015, received the Best Paper Award in the Annual Biotechnology Conference, Pune, received the Commendation Certificate from Chief Controller, DRDO Life Sciences, New Delhi, for First Responder QRT CBRN for the Commonwealth Games, received the Director Best Performance Award several times for untiring and excellent work in monitoring the various venues related to the Commonwealth Games, qualified the NCRC Visiting Fellowship from National Research Council of Canada, and received Srimati Annapurna Award from Society of Electrochemical Science and Technology, held at Annual General Body Meeting in the year 2005. Currently, Dr. Ponmariyappan is working in the TD project on design and development of biological point detection system, which is worth 135 crores. We are very happy, honored, and delighted to listen to your expertise, sir, and particularly on the biological warfare and how DRDO is handling it. Thank you, sir.
We request Dr. Krishnaveni ma'am to honor the speaker with welcome gift. Thank you, ma'am. We hope the participants are excited to listen the expertise of the speaker. Sir, the stage is yours.
Thank you, Professor Krishnaveni, for your introduction. I'm very delighted to be here. I thank the organizers and management of PSG to give me an opportunity to share my research experience with the young minds. Okay? So I'll just basically, I'm from Tamilnadu. You know, I'm from very in a village background, you can say. Where when I started my studies, we don't have a lights in our houses. We studied in street lights. You think of that today, what you people are having, you know. So we reached such a level. So the thing is there, you need to have an involvement, enthusiasm, and dedication. That only is requirement. If it requires to reach the highest limit, okay. So I'll start about, I'm from DRDO, you might aware, they introduced very well. So from the organization from DRD Gwalior. So I'll talk about few, what we are, yeah. So we broadly, you might aware that you know or no, I don't know. So that's why I'm just talking about DRDO. We have 8 clusters, but different clusters deals with a different type of areas. Okay, because you are amalgamation of all life science. So where I belongs to the clusters of life sciences. Okay, where we have 9 laboratories. So each laboratory deals with a specific area. So coming to this is for purely faculties. So what the life science research, we have 4 boards. These are the different 4 boards which I mentioned here. We used to fund to academic organization and research organization in this area. So we invite proposals and we used to fund it. So in that, one of the board is life science research board, where I am the we used to look for collaborations which of defense important, right? So what kind of funding we do is, you can name it the STARS, ERIPR. These are the 3 major names we used to fund according to the capital amount. For example, if it is 50 lakhs, 1 crore, 2 crores, 10 crores, above, like that we is categorized. Apart from that, we used to sponsor to always encourage the academia to conduct more and more exhibition, conference, workshops. We used to fund for them. And apart from that, now DRDO is one of the organization. We used to create a center for excellence to bring young minds, to give a exposure to them. You are fortunate you are having one of the center at Bardar University, Center for Excellence. So we have brought into a Center for Excellence. Earlier it is a Life Science Center, now it's become a Center for Excellence, where Dr. Kadirvel sir is looking after here. So like that, we started 15 more centers like that throughout India. Some centers are in IIT, some centers are in IASC. So you can go and they have a full-fledged research facility. We fund for them, starting from the infrastructures to research and the equipments and all those things. We give some fellowships also to the students so that they can train for the future research for the requirement of DRDO. Can you change? So this is the vision and mission of my laboratory. So we used to, DRD Gwalior is mainly for the nodal laboratory for the country. We work for the chemical and biological defense. We produce all the agent, all the scientists working for the state of was detection, protection and decontamination of either chemical agent or biological warfare agent. So this is our core competence in nutshell. We used to detect and we used to protect and we used to decontaminate. So and we used to provide trainings to the users, for especially the armed services, uniform people. Now we extended many more the paramilitary forces also as per the requirement. When country needs, we are always there to support. In COVID we did. We proudly says we are the only one in the country last 17 years we are giving the OPCW, Organization of Prohibition of Chemical Weapon Convention. We are the leader, we are consecutively getting A grade, and we are for the environmental sample and the biomedical samples. We very trace analysis within a stipulated time, we always get A grade in this. So apart from that, we provided a huge data to the OPCW. So we are the, what you can say, we prepare sample to send it to across the country, and those results will be evaluated at Gwalior, DRD Gwalior. This is a pride for the country. So apart from that, as per the requirement of the country, for example, the users basically, we need to protect, develop the technologies. We work for the 3 aspects: detection, protection, decontamination. We used to call the sense, seal and shape. Sense, shape and seal we used to call. We developed a technology for the detection. For example, if you want to, detection is foremost importance, right? The COVID says now, for example, when after COVID, you people are aware about what is this extent of biological warfare agent, like that. So according to the toxicity, this agent has been categorized. Which one will be biological warfare agent, which one chemical warfare agent, right? So the first and foremost thing is to detect the agent, then only you go for the next step you will protect. For example, a chemical agent, something got released, the gas and other thing you might have seen the recent Punjab cases, and the 15 minutes, hardly 15 to 50 seconds is required to get a symptom. In 5 minutes time, the death is, the agent like sulfur mustard has been inhaled. Okay, so exposed. So these are the detection system which we have developed. Just okay, some operation issue. Yeah, there's some operation issue. So okay. Okay. It's not moving fast. Okay. I get it. So these are the system which we for detection. For coming to the protection, for example, you need to, when something got released, a gas or a chemical agent, we need to protect the gears. We developed an NBC suit, we have the gloves, grounds, all those things. These are the items which have protect. We have developed at the DRD Gwalior. These are the portfolio. Apart from that, we need to decontaminate. We have developed a system, mobile systems, and we have shelters for mass, and we have a ship for NBC filters for different applications. We developed it. So I'll change it. Okay. Next, it's not working. So next is these are the different decontamination system which we have developed for to compact the biological and chemical warfare agents. Next, please. Just give a glimpse what are the technologies which we are working. So what are the products which we have developed? For example, when you look at the NBC market, nuclear, biological and chemical portfolio, 90% is the technology which was developed from DRD Gwalior, and only 10% we are importing. Okay, this is apart from the expertise which you talk about biological, what are the expertise which we have? We are expertise handling pathogenic microorganisms. You talk about anything, we have a BSL-3 facility. We are in the construction of BSL-4 is going on. The known pathogenic viruses or bacteria, any deadly viruses, everything we handle in our facility. These are the claims which we are doing. So apart from that, we developed a technology for detection system as well as vaccines and the neutralizing antibody for the toxin. These are the area which we working. If you go to any scientist, each scientist is working on a different one of the other agents. Somebody is working on anthrax, somebody working on plague, somebody is working on toxins, somebody working on virus, Marburg, smallpox, right? Like that it is keep on. So these are the detection capabilities exist with our system. So we have the systems for the ELISA and the PCR and the point of care devices for these are the agent which is already available with us. And okay. These are the virus panels which we have already developed the technologies. When the country, the COVID came, we are the one that, the first one we prepared the sanitizers, hand sanitizers. We are nearby Delhi, we prepared 2,000 liters and supplied to all the ministries including PM and other places. And we are the first one tested the PPE which was imported from other countries, whether it is working or not. So we tested a huge, every day we test hundreds of PPEs. Out of 100 PPEs, 10 PPE hardly will pass. More than 90% will fail. Because when they don't know, India don't have a system to test that point of time, they'll sell all those things. Okay. So when we rejected, then they started giving a good material to the country. So that we can save our life up. These are other systems, toxins, which we already have the developed technologies, which I'll be talking one of the toxins today in my talk. So next slide, please. Apart from that, we work on the society also with the vector control. Also, we developed the systems that is called a DEPA, cream as well as spray. When the armed forces are placed in the jungle, so there will be an open atmosphere. They need to protect from the mosquitoes. Okay, so for that, which has been developed. So it is already available in the public market or the civilian market. We have given a technology. I think one of the industries nearby Coimbatore also. They are TOT partners. Next slide, please. So another important area, this we have talked, the biodigester technology. Here, these forces is required when we deployed at the high altitudes, Leh and Ladakh. What they will do, a Ladakhi kind of toilets, they'll dig out the ice, snow, and use the shelter, and once it will be filled, they'll close it, then move to the next place. What will happen in summer, this ice undergo melting, that water will go to the pond, that pond water they are using for a drinking purpose, cooking purpose. So when it is mixed with the human waste, night soil, So it is, first of all, it's aesthetic nuisance. Second thing, it is prone for disease outbreaks. So there is a burning problem in Sciecnes and other places. During Kargil war, the deployment is more. So there is a burning issue. So that we developed a microbial consortium, anaerobic microbial consortium. Our team has visited Antarctica. We collected samples from there and we isolated a lot of psychrophilic microorganisms which is producing methane gas. So those has been prepared a consortium and which will work at low temperature. So that we made a design for that at different temperatures, psychrophilic, and we optimized the system. The technology has been developed, that has been bulk produced, and it is deployed in across the world, across India in the border areas first. Next, Indian Railways, similar problem. You have seen the railways, no? When the toilets were open earlier, okay, when you go into the railway station, you will see a lot of sit, okay, night soil will be there, that create a small bad smell and aesthetic nuisance also, okay. So the railway approached us, we developed a similar technology is converted into the various, because there the typical challenge is, a train is running from Kanyakumari to Kashmir. You talk about the temperature, in Kanyakumari it will be around 45, 40 in summer. Same time it will go to the Srinagar or the other places. In the end it will go, the temperature will be extremely low in psychrophilic. So we adapted the microorganism from the psychrophilic to thermophilic, okay, which will degrade. The hydraulic retention time of this train will not more than 4 days. It will reach there. So the complete degradation should take place in 4 days time. So this is what the cumbersome process, the task has been given to us. We developed a technology over a period of time and executed. We have done a 2 years time field trials across the countries, different trains. Then in railway, they may not change the train coaches. The available space they were given there. With that we designed a technology. With that we have implemented. Now proudly say, the budget, Government of India budget is when announced it is 12,000 crore. Most of the train is fitted with our bio toilets. Right. Next slide, please. So after that, the Lakshadweep Authority, the groundwater got contaminated. Tourism is the business. They told us, we need to convert your technology. We have converted as a different technology, the same kind of technology to them. So now they are having the entire Lakshadweep is converted to the bio toilets. This is what our story, what we did. Coming to the next slide. So coming to today's talk, what I am going to talk, what of the journey we started from one by one. I am taking one example, which we took the botulinum neurotoxin, where we started, what is the level in the preparedness in the country and what we reached there. So I'll talk, what is the botulinum toxin, you might be knowing. The previous speaker has talked about a lot of, very basic fundamental aspect in the neuro issues, okay, how he has addressed very scientific manner. So we like that in the neurotoxin, it is the symbol on each food, 1 spoon. What is the population in Coimbatore? So if I want to eradicate the population in Coimbatore, for me it's just 1 spoon is sufficient. Okay, this is a LD50. The toxicity of this toxin is 0.01 nanogram per kg of body weight, approximately. So one estimate was there, assuming a global population are 5.6 billion people weighing an average of 70 kg. So then I need only 39.2 gram of this particular guy is sufficient to eradicate whole mankind in the world. So you can imagine. So now you have seen cyanide, so many things now that we talk about chemical warfare agent. Cyanide is nowhere when you compare with the toxicity. The toughest toxicity is sulfur mustard and sarin and soman. These are the things in the king of chemical warfare agent. When you compare with the toxicity, these guys is requiring 87 tons, sarin is required 2.8 tons, 24 kg is required for VX, but in the case of botulinum toxin, only 2.6 gram is when you compare with the toxicity. So the issue why I am telling you, this is so toxic, people are very, fear to work on it. There is no vaccine is available, not in India, other developed countries also. Only the troops are given the prophylaxis, some vaccines and some neutralizing antibody. Not all the public has been not given. So people are not coming forward to work in this area. So it causes mainly, there are 4 types of botulism: foodborne botulism, wound botulism, infant botulism and inhalation botulism. These are the 4 types of disease, this agent which cause. So I'll be talking, so food, because it's a purely obligate anaerobic bacteria, when the absence of air, it will grow. So obviously when the packed foods, it is luxurious for the growth of this guy and produce copious amount of toxins. When you eat it, it will immediately cause flaccid paralysis. So when you talk about the international scenario, the detection system is very much available, vaccine is available for armed forces, and the antibody is available for post-exposure therapy. When you talk about Indian scenario, see, when I joined in 2002 for the DRDO, the scenario is the culture is not available, no detection system is not available, no vaccine is available or no therapy is available. Second thing, these are the few cases already reported in India, okay, sporadically. But second thing, we don't have any proper surveillance system. The cases is going on anywhere, most of the parts of the country, but we are not aware. So you look at the point care scenario. Next slide, please. Next, move it. Keep on. Okay. So when you look at both scenarios, scenario A and scenario B, you look at, in scenario A, when something comes, you have to test and result and you go for a treatment. In a scenario B, the thing is there, you need, for example, at the place, the point, for example, it's a field, when something is you can able to detect and you go for a treatment. But something is always not possible in the case of biological warfare agent, because we need to, sophisticated instrumentation, you need to go and to the hospital, the laboratory setup to confirm before giving a treatment, medical countermeasure. Because the medical countermeasure is very specific to the agent. And second thing, concentration, because it's have its own side effect. All the licenses, next please. So all the licenses has been issued based on emergency purpose, not for the routine purpose. So it will not go for the complete clinical trials. When you talk about that detection approach, we used the traditional approach, microscopy, serological tests, and rapid diagnostics kit, and we use molecular systems. Next place. But what is the limitation of these assays? These assays are poor sensitivity. Then standardization is difficult for this, and it is unstable. The reagents are perishable in nature, so it may not have a shelf life for a long time. So when I want to use this field, there is no electricity at all. How I can store it in refrigeration? That is an issue in field. Then always most of the sample treatment is essential, other issues. Then these reagents are very costly. Then in order to talk about botulinum toxin, these agents are, you want to do the gold standard is mouse bioassay. There were a lot of technical expertise required to use as an assay. It is time consuming. Next. So on the problems. So why, if you want to make a good one, what do you need? It should be simple and it should be small, it will be selective, very highly specific, and should be fast and versatility. Same time it should be user friendly, any layman can use, because the user is our 12th, 10th or 12th pass guys will be posted there. So I cannot go and every day teach them in border, right? So they need to do it themselves, and accordingly they used to take medicines also themselves. That's important. They will one point at a time, the soldier, they will do both the jobs. He will be, sometimes he's a clinician, sometimes he will be act as a doctor. And this is really required in field. Next slide, please. So what are the approach? We don't have, the country don't have a culture, country don't have anything vaccines. What we do it, we need to do it, we need to detect the agents, and we need to make a protection, and we need to, something got already exposed, we need to neutralize antibody. The three areas we want to work on it. The approach we use, we developed a molecular system first, and we followed by the immunological. These are the assays has been developed in the course of time. Then these are the approaches which we use, the protection subunity vaccine which we developed, and these are the antibodies which we developed. I will be describing in my subsequent slide. Next slide, please. When you talk about, next slide, please. When going for the toxins, for example, detection, what we did, the country doesn't have these organisms. So I was there, I worked with Bureau of Microbial Health Hazards in Canada. So where I worked exactly with this particular agent in the director. So we put a lot of simulation tests over there. They used my expertise, and after coming back to India, we, from south to down south to the north, we collected sample, possibly the butcheries, where the anaerobic stops will be there. So we collected that and we started isolation. Fortunately, today the country is having 55 isolates of different subtypes, and we have 3 of novel serotypes. So we identified these, the very cumbersome process to work in these, because a molecular level of oxygen will be there, these guys will sporulate. That's the biggest problem. So we need to work in the glow box. It's an anaerobic chamber. One of my students is working there. The typical cardiomyology is a spore former. Some oxygen goes, it will convert into spore. First we optimized with the agents and 16S rRNA based identification. Then the toxicity by mouse biopsy. Then we developed a PCR, which something comes, we can do it easily and kill the sample and extract the DNA, and we can do it, test it, which type is there. So since we don't have vaccine, so we cannot expose the toxins. But we also have a psychological trauma. It's not like that. When I am working for a long time with this agent, it is have a pungent odor, this anaerobic gas elevates. Okay, so sometimes it aerosolize, when your opening will be there, it will aerosolize. Something I feel, I inhaled some toxins. So that's why I'll be there tomorrow or not. I used to tell. So this is a trauma mentally. When after that we come to know this not happens. That's why we started working on the molecular aspect. It's pretty easy to do it. Next slide, please. So once we developed this individual PCRs, next. It's moving. Yeah. What we did, we developed a PCR, and after that we developed a multiplex PCR. So individual PCR putting is a problem, every time they changes the result and the sensitivities keep on changing. At different matrices, it is keep on changing. Have issues changing? Okay. So what we developed a multiplex PCR so that we all put together all A, B, E, F, because it is 7 serotypes. Out of 7, 4 is responsible for human botulism, and another 2 is for aviation, animal, these birds, and it will cause botulism in animal. So we developed a multiplex PCR for A, B, E. You can able to see these things. So you can a single go, you can able to see whether this guy is there or not. So you take a soil sample, you suspect, you take a food samples, you are suspecting, you take it and simply you put a boil, you just boil the sample, and this supernatant you load it, and you can do a single PCR, simple PCR, you can able to identify. So of course but the thing is there, the detection is easy for us. We have completed and we say our authority, we could able to detect it. So what next? They told, okay, it's fine. The question arises, I want to detect a toxin, not the gene. Okay? So these bacteria might be present in everywhere, right? So it will be in the it will be present in the environment, in the soil, it will be there, it will nothing to harm you. So when it is anaerobic condition comes, only it will go sporulate, and it will go inside, and it will convert the vegetative body and produce toxins. Ultimately, the molecular detection doesn't help us. So we don't know that. Okay, but something we did, then they told we need to detect active toxin so that we don't have a culture. In the meantime, what I showed is that point of time we don't have a culture. So later on we identify isolated. So what we did in 2004, they started a synthetic biology. So we know the gene is very much available in the PubMed, the toxin gene. So what we did, we designed a PCR by using a PCR overlapping method. It is a cumbersome process, but we did, and we synthesized the toxin gene. It is 1400 base pair for all the 4 types. So after that, we cloned in a vector, and we artificially produced a protein when we generated artificially, and we expressed it in an E. coli, and we produced a biologically active protein. This is the expression which you are showing, 55 KDA protein, which is good expression. So then we purified, we attempted with native condition as well as denatured condition. Fortunately, we try to purify in native condition, so that we need to have the surface recognition, so that it should not be denatured, so that you can able to, when you are developing an antibody, this antibody should have a both detection purpose also we can use for the neutralization purpose. So this is the system we expressed, and we confirmed with the antibodies. When we purified the 90% homogeneity, we optimized the different media, all the stuffs, and we got other things, the immunization, and we ultimately, it is able to give protection. You can able to see with the different adjuvants, and ultimately this particular antibody is highly specific. The titer is 1 is to 32 lakh thousand, so it can able to give a protection. So the detection system, the detection, it is highly specific with along with other cross-reactivity. Similarly, when we are immunized with the different adjuvants, we don't know which adjuvant is good. Then we have seen we have got a very good titer in the case of alum. So that will go for the next interrelation assay. Next slide, please. Next, skip on. So similar whatever we did for the different serotypes. So once we have this titer which I showed there, so we purified the IgGs and we optimized the immunodetection system basically for ELISA based system. So once we detected the detection limit is 7 to 7.5 nanogram per ml. So once we detected, so then we spiked with, we don't have the clinical sample with us, we spiked with these different food matrices and we evaluated against the system assays. So these are the different detection limit for the different matrices which we have shown for the type B and type E which we have shown here. So the detection vary from roughly is 50 nanogram to 120 nanogram. It's there. So these all we have already published. Next slide, please. These are the things which, so once we did all those things, the pretty good, we can able to detect large in number in ELISA at the laboratory is fine, perfect. The next question they asked us, okay, it's fine. Will you able to detect in field? We don't want, nobody wants to come to the laboratory to always test because the forces are deployed at the borders. They need to detect there. So the question arises whether whatever the antibody which we developed against this 4 serotype which is amenable for field applications or not. Immediately we go on for the dart ELISA. Similar approach, we optimize the dart ELISA. Yes, you can able to see it, the detection limit is 31 nanogram per ml. Similar different matrices we did in whether it is alone it is detecting, then it is the matrix it is detecting. So both is work. Then we put the flow through assays. We optimize in the laboratory. Yes, pretty much it's good. It is able to detect the flow through assays. Then next slide please. What next we did? Simply it should be stability. These assays won't be stable. So ICT will be more stable. We gold conjugation we did this assay. This typical ICT which we are showing one is. So it 50 nanogram sensitivity it is there per ml. So which is able to detect. So what we did when we pulled together 4 ICT differently for ABE, then we have given to services so that readily go for the deployment. So the thing is there when you talk about which I showed you one slide earlier, the toxicity of this toxin 0.01 nanogram per kg of body weight. But what I achieved is 50 nanogram, which is very, very less. So you cannot able to see. The next question they ask, what is the LD50 of the system? So what do you have? You need to increase the sensitivity of the system. So these assays have its own limitations. Okay, we moved on. Next slide, please. So what we did, we used a lot of chemical, this thing is mass spectrometry. We used a different approach. What different approach means, whatever I showed is a traditional, but we go for the molecular mechanism of these toxins. These toxins, what will happen now? It is neurotoxin. Basically, it is useful for the contraction and relaxation. Okay? What contraction, muscle contraction, relaxation, what is responsible? Acetylcholine is responsible for the contraction, relaxation. What this guy does, this toxin it will go and cleave the snare protein or the VAM protein, depending upon the serotype. It will block the acetylcholine synthesis. Once there is no acetylcholine is there, the muscle will be always in the relaxed state. Okay, it will not contract. So that property we exploited. We took the snare protein, the region which I mentioned here, we put the toxin, it is cleaving or not, we expected with mass spectrometry, this method works, it's instantaneously. Similarly, different matrices we did, so we got a clue from that. What next we did? Next slide, please. Once, yes, this method works, so we can go. One thing is there, I can able to tell you, how many of people, you people are seeing movies every day nowadays, why you look at always the heroines get more attractions always in any movie, right? How a 50 years a lady looks like a 15-year-old. See, all our heroines look like pretty young. Have you seen? Are you agree with me? No. Agree? Yes. Then why they are looking young? Have you seen anyone have answer? Good. They use a Botox. What is Botox? It is nothing but a botulinum toxin, right? Just I told, the fundamental principle, mechanism of this toxin. It always, when the toxin goes, it will cleave the synapse protein, the muscle will be always relaxed state. What will happen when aging is going on? The previous speaker told very nicely, okay, whether you catch or not. When aging is happening, what will happen? You will become wrinkles. Shrink ho jata na? That whatever the face, all the places, your muscle will get shrinked. When shrink is there, you inject the toxin in a low dose, 1 unit or 0.1 unit, in that particular place where the wrinkle is more, shrink is more, what it will happen, it will relax the muscle, right? So it will muscle relaxation, you look like fresh, so that there will no wrinkle will be there. So that's why most of the heroines, they undergo for a Botox treatment. So that's why they look like always young. Okay? So it's very costly affairs. It needs a lot of expertise to know how to give an intradermal injection, right? It's minimum 10 doses is required to different places. Okay, you can see in the movie now it is already available. So once we have that, in the meantime, parallelly we isolate the organism and we look for the vaccines to be developed when some forces is going to the field before they should be immunized. So we need to identify the markers which we can use for the organism which is giving. So what we did, we immunized the organism, we grown the organism in different media, and we look for the proteome profile, 2D proteomics, we immunized the whole cell antibody, secretome as well as the whole cell protein, and we elucidated the different function of the particular which we listed, and the reactive antigen which have worked out, which are the possible markers. So those markers has been again that has been amplified recombinant and produced, cloned and expressed, and antibody has been developed and look for the potential. We have tested in mouse model whether it is giving a protection or not, and challenged with the toxins as well as the organism. These are that same time which has been used for a detection also. So we could able to identify the recombinant OTC has been as one of the potential biomarker for this particular guy, and there are a few markers, GROL, which is specific to some serotype which is giving the protection. Next slide please. So these are the in vivo, next slide please. Next. So these are the assays which I showed earlier. These are very earlier assays which I showed, 50 nanogram which we reached the ICT, which is very less sensitive. So in order to increase the sensitivity of the assay, what the approach we use, we collaborate with the material chemist, material science people. So we use the signal enhancers. We use a biosensor approach so that you can able to increase the sensitivity. How? The surface areas will be enhanced. The more and more molecule antigen will attach to the antibody, so the sensitivity will get increased, and the signal will be amplified manifold. You are using some signal tags. So that will be using some few slides I'll show. Next slide please. Next. So here there are some materials which use the graphene nano seeds which have been used for as a signal enhancer for that. Next slide please. So apart from that, you are all aware with the gold nanoparticle has been commonly used for the detection purpose, right? So we prepared the different nanomaterials with the different size. Which size will be optimal for the detection, which we find out it's 40 to 50 nanogram is very optimal, and it will give very good signal for this guy, and we optimize. Next. So in order to, in a single, next please. In order to increase the sensitivity, for example, it's have its own sensitivity. If you are using a gold as a tag, gold will be tagged with conjugate with the antibody, and it will be enhanced. You have a 10-fold increase. Still I have 0.01. I may not reach that. So that this is the detection assay which use the sandwich assay using electrochemical approach. You have a simple sensor, you can put the drop and you can check it out. So signal enhancement you can able to detect. From 50 nanogram to 10 nanogram I reached. Next slide. So further enhancement, these are the detection limit. You are able to see this particular guy is using for the one particular serotype E. You can able to see, we can able to differentiate, but not 10 picogram it will go like that. Okay, next slide please. Further a different approach we use. Here what we did with the boron nanonitride, boron seeds has been signal amplifier we use. We use the 3 molecules tagged one signal and another tag and third tag over the whole tag, and they capturing, revealing antibody. So whole bunch will be used here and much more. So the next step please. How this is the approach you can able to see graphically represented. This boron nitride seed will be there. That is a signal molecule. There we'll coat at top of that this gold nanoparticles. So you'll have a two-fold increase. The 20-fold increase will be there. Beyond that, we have coated the antibody which is specific to the botulinum. Similarly, the approach B here, the same thing. There's a one punch. Generally, we are revealing antibody will coat with 1 nanomaterial. Here we put 3. Like that, we put the similar. The antigen will be coated in one. Here you can able to a sandwich able to detection. Here you increase much more because it's have its own thing. Next slide, please. So still we may able to increase. The research, they never ever, satisfy with what you achieved. So always a good researcher will keep on moving to go hard further, right? There will be no never-ending stop. Next slide, please. So what we did, another thing is there, the invent of quantum materials come into place, a lot of literature published, so you can increase much more sensitivity when you are using a fluorescence. Always fluorescence, the limitation, it works very fast. Okay, the interaction will be very fast. You will get light faster, then the signal will be much more higher. So that we started using quantum material to further the developed antibodies will be tagged with that. We can switch off system. Next slide, please. If we can use the cadmium material, we have used this kind of nano quantum rods, cadmium based, and in that over we loaded the tellurium. Beyond that we tagged with a particular antibody. With that we use, whenever you use H2O2, there will be a turn on off. For example, specific interaction will be there, that fluorescence will on. If it is very specific to the antibody which you want to detect. That approach we use. So we got a very good system that is field amenable. The earlier one, which is not field amenable, it's an electrochemical system. You need an instrumentation. Here only just a reader, fluorescent reader. It is common for a single reader, the single monochromator based. It is possible to do it. So that instrumentation is much more easy. We approached this. Next clip. We reached this 1 picogram sensitivity. Initially we approached 20 picogram. Then if we did a slight modification in the process, we go for a 0.1 picogram per ml. Next clip, please. So further, the same assay which is equal, we moved to the surface plasma enhanced Raman spectroscopy. The SPR based detection, which is highly sensitive in that. So we moved to the SPR based system, which will be very useful in the laboratory, not in the field, because we have two systems. One is on-site, one is off-site. Some of the things we knew to the field. So, but always in a military system, we need to have two different technology. They don't believe on single technology before declaration. You need to develop two alternative technology. Unequivocally tells, yes, this is particular agent. Then only you will declare, commandant of any unit commandant will declare it. So that in order to develop this guy we worked it, so we are able to detection limit is 0.1 picogram per ml. So next slide please. So everything is fine. We developed everything is fine. We reached the sensitivity what we want to the LD50 of the toxins. Now the question comes, the COVID has come into picture, because before COVID everybody is okay. After COVID, what they told, nobody want to go to the hospital. Nobody, no. If, for example, doctor may not be interested in initial first wave 1, wave 2. COVID wave 1 and 2, no doctor is going to the hospital, no hospital is opening, because they have a fear. Okay? Now, if a doctor is having a fear, a clinician, how he will come forward to test? So, that is the biggest problem, because we are the one, we have a BSL free therapy. All from Delhi to Gwalior, they will move the sample 5 hours journey, they will send the sample to us to detect it. Period. We tested first. So, then the autonomous detection has come into place. What we are doing, we developed a wonderful technology. I can able to share you some of the things. So some of the strategic areas I cannot able to, we are working much ahead. I may not be able to share those things to you, but I can, some open literature things I can share you, which we have done. Next slide, please. So, see, there are some of the techniques which is important. So there are some aromatic acid biological molecule which is present in every body, the biological microorganisms, viruses, and some of the bacteria. So there are these aromatic amino acids are in fluorescent, intrinsic factor is fluorescent in nature. So when you excite a particular wavelength, it will absorb and it emits a higher wavelength. That is a lambda max, the fluorescence, we extended wavelength. That particular property has been used for to have a early warning purpose. What country like India needs, we cannot deploy PCR machine, we cannot deploy all those costly equipment. How many you will put in? In a city like Coimbatore, how many hospitals are here? How many instruments is required? We cannot put 1 crore or 50 lakh instrument in every place. So we need to go low and cheaper systems, and it should be consumable free. I can able to give a PCR to you, but what about real-time consumable? Again, it is day-to-day basis. There is no case, even then you will use it, and if it has a limited shelf life. So the system which we need, country like India or the developing countries, the low-cost instrument, which the consumable free, right? So this is what the technology, this particular phenomena we have used. These are the aromatic amino acid, the absorption which I showed, the emission I showed. Here we used a particular wavelength to develop a system instrumentation. We did in 2 years time, the only 3 countries having this particular technology, only 2 people or 2 countries is developing and supplying across the world market. We are the 3rd one developed this technology within 2 years time in COVID. Next slide please. So this is the project which we are having, the architecture which we are using. It will collect aerosol continuously, air will be sucked, it will detect, and there is an elevation in the biological load, it will give an early warning to you. The response time is 30 seconds. You can imagine, 30 seconds. But we are giving, it will work on nanoseconds. In 0.1 nanosecond, the system will real time, it will measure, but it will give an average, and we put a AI here, and it will give within 30 seconds, it will give an early warning to you, alarm. So something has come in. So what we'll do the next case, the biosampler is attached to that, automatically it will actuate the biosampler, the sampler will collect sample and it will concentrate more sample. Why it is required? In environmental, the agents are in much diluted form, you cannot able to detect, there is no technology is available, directly you can identify the agent from the environment. So it need to concentrate it. So here it automatically actuate the sample, the 10 minutes it will collect, 6,000 times the sample will be concentrated, it will bring into the vapor, air phase to liquid phase. Here it will come, we shall go to the immunological. You might have seen the pregnancy strip, the ICT which I showed you. The array of ICT will be there. Automatically the sample will belong to each ICT automatically. In 5 minutes, 1 minute after that, it will be, a card reader will read, and it will barcode it, it will tell what is the agent is present. The whole duration will take in 5 minutes. In total, you are taking 30 seconds, 10 minutes collection. Okay, exactly you will have 11 or 12 minutes, you will come to know what is the agent is present in there. Here there is no expertise required, there is no manpower is required. But you will get a early warning in 30 seconds. So once the early warning is there, in few places, normally the critical infrastructure which are available in the country will be run with NBC filtration system. So that normally it work on the dust mode, it will actuate the NBC mode, the air which is going to the building, it will come through that, you will get protected. So that is the idea, whole idea. Once it will be done, we don't believe, I told you, we need 2 different technology. Though 5 minutes we will come to know, so that it will go automatically, the sample will go parallel approach in a molecular system, real-time PCR for agent which is specific. A DNA will be extracted on board, and it will run it automatically, and it will tell what is the agent and is the concentration. Then only the threat will be declared, it will be confirmed. We earlier will provide early warning, and it will be confirmed. Then the person is going for a medical countermeasure. He will be given with a tag, the patient got exposed this agent with this concentration, so that doctor easily will give the medical countermeasure. There also we develop a medical countermeasures first aid kit, so there the doctor we are giving easily, so that this is the organism, this is the dose, it will be there, you can give directly. So what we did, the early warning device which we designed approach, we designed, next slide please. With Bell is our industrial partner. You look like this is the instrument which we build it, the inside with the outside with, these are the toxins. We want to focus on the toxins. Why? It will detect all the agents, it is detecting, but here the response time, you can see the average response time which I shown there. It always, when you expose to these biological agents, it will give a, yes, warning it is giving in 30 seconds of time. Next slide please. So next, these are the identification system, which we have developed, and the moleculars, earlier the immunological and the molecular one which we showed there. This part is pending here. This is the approach which we are using here. This is pending currently. It is under development because the assay has been optimized for here. The manufacturing is done in India. Next slide, please. So what is next? There are some areas we have gone much ahead. I cannot say here what we are doing next, what we are working on it. We want to don't go for the instruments. This is okay, fine, autonomous, everything will monitor in the border, everything. Some soldier is going somewhere, so the sensor should be placed in their suits or the wearable sensors. All the agents, the sensor will be miniaturized, which we are collaborating with many people. We are funding also with the wearable sensors. The agents, whatever the chemistry which we developed, that will be converted into these, and we are working on that. Something got detected, will give different colors of light, so that, for example, it is yellow color, it is anthrax, it's green color, it is toxins, like that, botulinum toxin, like that. So that automatically we have developed auto injectors. The drug is filled with in a cartridge, so it will be in the thigh automatically. For example, it is green, so that a soldier can act as a doctor. He need to just press the thigh, so that the particular green auto injector, the drug will be injected, he will saved. So you need not to worry. This is what we are working. Next, see the other area which we are already working, our point. We want to detect much early enemy is using in the border areas. We don't want to take risk, so that somebody is releases something in the border, for example, 1 kilometer or 5 kilometer distance. So some cloud is moving, some chemical or biological. So using this for LIDAR technology, we used to capture the signal, and we used to be able to detect whether it is an agent coming towards you is a chemical or biological. If it is chemical, so we used to send, we have a drone system, we used to send and collect the sample, and it will come to the station where you can able to identify what is the agent is present, but it will provide a early warning before much ahead. For example, the current we have developed a 1 kilometer range with a 360-degree rotation with 20-degree up and down. It can able to detect. What we are working now, it is 5 to 10 kilometer range which we are doing it, this early warning purpose. So for example, we have a drone up to 1 kilometer range, you can able to collect sample and for confirmation. Next slide please. What we are next, this is a health conference. The idea is what we are in the armed forces, what we are using, somebody is there, for example here I put some modified pictures for this conference. So what we are looking for that, for example, the future autonomous disease detection, somebody, something, for example, a patient, his heartbeat is, the previous speaker told how the cardiac thing is happening. You may have seen cardiac arrest is very difficult, you need to reach the hospital. Before you reach, the clinician will be there ready with all your symptomatics, because your database is already there, so you can able to treatment and you can save the life of the people. This is what exactly we are using. We are mimicking with warfare agent in army scenarios. We are already adopting and we are using it, this technology. But my expectation, this is workable solution. You can, because some modernization is required in the hospital setup, and it is very much feasible also. My next slide please. So this is the ultimate requirement, the future smart home. For example, you want a home, because in the developed countries what they are looking for, you are a home built with everything is there, your home with all the sensors. For example, they have a lot of energy saving also in the system nowadays. Already most of the houses people have started using, so because they are not going to switch on and switch off, because the sensor based, if you put the sensor, when you go in, automatically the light will on. If you go out, the light will off. You might have, many of the people have seen those kind of things. So like that, you can operate, you can monitor the devices, sensor will be placed. When you are not, for example, if you are in bed, for example, you are having some breathlessness, illness, automatically you wear some smartwatch with have some features that will be attached to the GPS. Automatically, you need not to worry, you may not able to call. Automatically, you give alert to your clinician, he will come and pick you up and your life will be saved. And some theft is going on the similar way, something unknown person is coming, there are a lot of face recognition software you can put it, so that these are my family people, these only automatically the door will be opened, other people comes, automatically a threat alert will be given to the police, so that they can easily catch hold of the theft people. Okay, these are the future India. Okay, thank you. Where we are.
Thank you, sir, for addressing the various scenarios of biological applications in defense sector, besides elaborating the various detection assays, all of which would have given various glimpses of research ideas and insights to the audience. We now kindly request the chairperson, Dr. Naren Ramanan, to outline this technical session.
That was a very interesting talk given the times that we are living in terms of both chemical and biological warfare. What we need is things like these, the most important detection systems, aerosolic and in all forms as well, in fact. And the best institution to run this is nothing but our defense systems, the DRDO funded research. And this is at the cutting edge of where we should be going and what we should be doing. And I hope you all appreciate, given what we went through for 2 years, 2021, in terms of COVID. If we had had better detection system biologically, then it would have been very useful. But this is very, very serious. The chemical warfare, chemical reagents, especially coming from bacterial systems. And that was very, very informative talk. Thank you very much. Opening the floor for questions. Please feel free. Exciting. You can come up with anything that you want to ask.
Yes. So we have heard of this point of detection on E. coli and others, which can be detected easily. Otherwise, we go for a lengthy detection for E. coli in water, particularly for the contamination. So, like botulism, the toxin identification, is there anything that you've done on E. coli for water quality testing? Is there anything that you had done?
See, given an example, to lake up time, there are many things we did, we were not, lake up time I couldn't able to explain. Your question is right, because we have developed the first aid kits, we have a water poisoning detection kit. There, you want to go, you are going to the field, you want to have this water is drinkable or not. Because the enemy, you are moving towards crossing the border to other nations, you may not know they will be filled with toxins or something poisonous, cyanide or kind of things. You don't know. So they'll move with the water poisoning detection sheet. There is simple method. You just take a paper and you dip it. There you will tell the color based on that the water is pure or not. Number one. Number two, you'll move ahead, you cannot carry water over there. The water which is available might be dirty. So you have a simple, there is a straw system, you can put, using the straw, for example, contaminated water, using the straw, you can suck it and all the water will be purified with all the contaminant. The pure water will go, you can drink it. That kind of system we have developed. So, coming to the area which you asked, E. coli, not only E. coli, there are several organisms, salmonella and so on, many organisms. We have antibody-based system is already with us. Anybody interested, we can try to help. If somebody is interested to work on this area, we ready to fund also. Because you may not really work on warfare agent, it is not possible, you may not have the facility, but these are the technology which you people are interested, we ready to fund, you work on some simulant. For example, we will provide the simulant, you develop the system, then you come back to us, we will provide the laboratory space for you at our place to validate your assays with real warfare agents. With a chemist or biology or anyone, we really welcome. Thank you. Thank you.
Any other questions?
Good afternoon, sir. I have a question here. So after the onset of this COVID, we all know that there are more pathogenic organisms in the upcoming future. So do you have any strategies to develop or identify such an organism before the onset of the disease? As we were working on the botulism on all the organisms, it was the outburst was maybe more than 3 decades ago. So do you have any strategy to identify an organism which is...
Yeah, it is very much possible in the ophthalmology. So you can do it. Because see, the technology is again, it's common. Where you want to use, which platform you want to, you need to select. What are the things there? Whatever I told, different technology, because we are not mastered in those days. Okay. But that is the need of the hour. You have to educate and you need to pick up what is available commercially, the technology, what you try to learn and develop it. Like that, accordingly, any requirement. For example, this is the technology is common. According to the application, you have to mold it. It is very much possible.
Okay, sir.
Okay.
Thank you.
If no more questions, let's thank the speaker. One more question.
COVID. So, and different serotypes of the same virus or bacteria itself. Yes. So, if we set up a system like we have a sensing system in a building for different bacteria and all the bacteria and virus till we know till now. So, we can, for example, if this building is have these sensors and a person enters this building with COVID-19. Yes. Okay, sir. Yes. So, when he sneezes or something, the virus will enter into this building's air in the form of aerosol. So, if we sense it in picogram level, a computer can give us the information, right? This area is contaminated with this particular virus or this bacteria, which is infectious or contaminous in this concentration, and this information can be given to the hospitals or hospitals. So we can tell, right, we can isolate this place and we can tell some contagious virus is spread over here. And this is one concept, sir. And if we go more advanced, I don't know if we can isolate a DNA from this. If we are able to isolate DNA from aerosol, then if the DNA is integrated with Aadhaar, so we can identify the person itself. No, sir. If we have the number, we can inform him or her that you may be infected by this virus or bacteria, so you can get the diagnosis. So can we do a setup like this?
Yeah, very much possible. I can tell you it's a wonderful question. See, the thing is there, you talk about, I just given an example, that's why, for example, it is indoor monitoring when you are using it. For example, the trigger system, the laser-induced fluorescence is a common. It won't discriminate. It will cater any damn agent, known or unknown. But it doesn't discriminate between the virulent and non-virulent, pathogen versus non-pathogen. It won't discriminate. But there is a raise in the biological load. It will provide a early warning, right? Now, you will have an alert. Somebody is coming, entering inside, something alert is there, the person is having some problem. Okay, that you need to identify, right? That will be there. So what we are using, because there are strategic system, we use a different approach. Somebody, for example, this room around 200 people are sitting over there. Somebody, we will temperature profile map, we do it. Okay. So my cameras will be there, the top, it will be temperature profile. Automatically, some guys is having a temperature. There are 2 people in this room. Temperature is more than the body temperature beyond the threshold. Their light will be flashed there. These people will be picked out automatically from our armed forces. They don't ask you, they'll come and pick you, they'll take you to the hospital. You cannot escape. That system we have, right? Number 1. Number 2, yes, somebody is coming, you have identified. So the person is coming, it's sneezing. You can go for a PCR and other thing for all the agents you can do. But it's a cumbersome process. Nowadays, people are not ready to give blood. Now, non-invasive technologies are gaining more importance. What we are working on it, we have developed a Raman-based identification. We are generating a signature, Raman signature for each and every bacteria, okay, all those pathogens, public health importance also. So, there you can easily identify, okay, for example, simple sneezing, somebody, sneezing, from that you can collect and do it. You can have the saliva, from the saliva you can identify. Blood, of course, blood is also possible. It's easily you can doable. So that you put a 1 drop of any of the things, and the Roman signature will be generated in 1 minute time. The maximum time is required for 0.5 second. So here 1 minute I am asking double the time. The processing also in 0.5 minutes. The library is all inbuilt for each pathogens. So it will go and search the library, fingerprint match, then it will tell what is the organisms. You can able to do it. This kind of system is already we are developing, but a lot of validation is required. We are in the process of validation for biological warfare agent. The same technology we are going to give it to the Ministry of Health also for hospital setup, but there a lot of data is required, pre-data for the pathogens with the different temperatures, because the fingerprint, because the same agent with the validation, we need to have a huge data with the different temperature, different humidity, all those things, fingerprint will vary. So that validation is going on. Some of the warfare agents concerned, but when the technology matures, it will come to the market. It will go to the transfer to the industries. It is possible. Okay. Thank you, sir. I hope I might answer your question.
Yes, sir, you answered.
Thank you, sir. We, on behalf of the organizing committee, invite Dr. Valarmati, Associate Professor, Department of Biochemistry, to honor the speaker with a memento. Thank you, sir, and thank you, ma'am. Always have an attitude of gratitude, says Sterling K. Brown. We hereby heartfully express our gratitude to the speaker, chairperson, and co-chairperson for engaging in this technical session. We now request all the delegates to proceed for a short tea break and to please join the technical session 5 at 5 p.m., which will be followed by cultural events and dinner. So kindly assemble in the auditorium by 5 p.m. after the tea break. Thank you.
and the respectable chief guests from Sri Lanka and South Korea. Good afternoon to all of you. I am immensely happy to introduce the chairperson, Dr. Kalpani Ratnayake. At present, she is the deputy head of the department and senior lecturer, Faculty of Health Sciences in Sri Lanka. And she is an enthusiastic scientist, researcher who possesses a substantial knowledge and experience in the field of biochemistry, laboratory animal anatomy, and natural product chemistry, and she is having passion of teaching. And she has got a wide professional experience as a researcher and as well as a teacher. She has contributed a voluminous details about the subject for the UG degree students and PG degree students to get the degree and their development. And she has got a very good curriculum development as well as in various scientific societies, national professional bodies, and she is also an editor and also a member in the editorial committee. And she has been invited many times to be the head of the scientific conferences and technical sessions in a very good manner. And she also a resource person for various programs in her country and abroad. And she has also acted as a judge for the various activities in the technical sessions like today. And she has also chaired the events and notified the prize winners. And she is also a reviewer and for the various journals of national and international levels, and she has won many awards. And she has published many research articles to her contribution to various journals of national and international levels. So I have taken the extract of her contributions to this level. If I try to speak or talk on the real data that he has given to me, will take more than an hour. So in order to minimize the time, I have given the gist of the details of her contributions to the Society of Science in her country as well as abroad. And I feel very happy and thank you. Good luck.
Thank you, sir. Now we are excited to listen to the presentation. Ma'am, the stage is yours, please.
Okay, thank you very much for your kind introduction and good evening, dear all. So my speech is based on identification of anti-inflammatory activity of natural products adhering to the 3 R's in laboratory animal science. Next slide, please. Okay, so before that, I will give a very brief introduction about my country. I came from Sri Lanka and you can see that Sri Lanka is rich with biodiversity and the natural beauty. So I'm inviting all of you to visit one day Sri Lanka. Next slide. And here you can see some of the photographs of my university. In this side you can see the photograph of University of Sri Javardhanapura, which I pass out. And here you can see some of the photographs of Sciecnes campus Sri Lanka, which I am currently attached. Next slide, please. Okay, so with that introduction, I will move to today's my speech. Next slide. Okay, so inflammation. I hope all of you know what is an inflammation. However, I will give a very brief introduction. So inflammation is a pathophysiological response to injury, infection, or tissue destruction, and it is characterized with 5 cardinal signs. Here you can see the redness, then heat or a warmth, then edema or swelling, then pain and loss of function. So those are the 5 cardinal signs of the inflammation. And here you can see the importance of the inflammation. So as I mentioned in earlier, inflammation is a body protective mechanism, and it is important to localize the causative agent and limit damages. And also it is important to remove damaged cells and tissues. And when we are talking about the causes of the inflammation, there are some endogenous causes and exogenous causes. So when we get these exogenous causes, there are some causes which come from environment. So you can see there are physical agents such as heat, cold, radiation, mechanical trauma, and also some chemical agents like inorganic agents or organic agents, and also biological agents such as bacteria, viruses, or any other organism. And when talking about endogenous causes, they arise within the body. And here you can see some of the examples like hypersensitivity reactions, which are antigen-antibody reaction, and also some enzyme releases such as proteases. Okay, and when we get these inflammation, depending on defense capacity of host and also the duration of the responses, we can classify the inflammation into 2 types. So there is an acute inflammation, which is a short duration one, which can last in less than 2 weeks, and also chronic inflammation, which are longer duration one. In here, the chronic inflammation can occur either after causative agent of acute inflammation, persists for a long time, or the stimuli which induces chronic inflammation from the beginning. So why I am telling these 2 types? Because when we are screening anti-inflammatory, the compounds with anti-inflammatory activities, there are certain tests which are target acute inflammation as well as chronic inflammation. So according to our requirement, we have to select the correct model when we are screening anti-inflammatory action. Next slide, please. Okay, so I mentioned that inflammation is a body protective mechanism. So why we are worrying about anti-inflammatory action if it's a body protective mechanism? Although it is a body protective mechanism, when it's happen in excessive manner or uncontrollable manner, it can make certain damages. So that is why this anti-inflammatory action also very important. Next slide, please. Then why do we need new anti-inflammatory compounds? Next slide. When we get the available treatment for the inflammation, the most commonly used drugs are steroids and non-inflammatory steroid drug, the non-steroid anti-inflammatory drug.
which are commonly known as NSAIDs. And when we get these drugs, these drugs can make certain adverse effects. For example, some of NSAIDs, they can make gastric irritations, which can make gastric ulcers. Because of this reason, nowadays the research are mainly focused to find new anti-inflammatory compound from plant materials. So in that scenario, these natural products are very, very important to us to screen new anti-inflammatory compounds, or we can incorporate these natural products into different formulas which we are hoping to develop for the anti-inflammatory action. And when we work with natural products, first we have to check that particular activity for the crude extract. I hope some of you may know there are certain techniques that we can make the crude extracts, like the reflux technique, Soxhlet, macerations. Likewise, there are so many techniques, and according to our requirement, we can select the correct technique. And if we are checking one of the interest activity, first of all, we have to check that activity for the crude extract, and if we see that activity in our crude extract, then we can do the fractionation, as you can see in here, and then we have to screen the fractions, and then we can select the most active fraction, and we can work on that, and finally we can end up with isolated compound. So that is the normal story behind the natural product research. Next slide, please. And here, when we get these inflammatory responses, there are vascular events and cellular events. These vascular events can be divided into two types: the hemodynamic changes and altered vascular permeability. And the cellular events may be due to exudation of leukocytes or phagocytosis. Next slide, please. So when we come to these hemodynamic changes, here you can see some of the steps in these hemodynamic changes: the transient vasoconstriction, then the persistent progressive vasodilation, elevate the local hydrostatic pressure, slowing or stasis of microcirculation, and margination of leukocytes. So when we are conducting research, why this background knowledge is important to us? When we are screening the activity, we can focus one or more of these steps. For example, here you can see when there is a vasodilation, it can increase blood volume in a microvascular bed of the area. So it is responsible for redness and the warmth at the site of acute inflammation. In earlier I mentioned there are 5 cardinal signs of the inflammation. So when we work on these anti-inflammatory action, we can target those cardinal signs, and we can screen the anti-inflammatory activity depending on the mechanisms which are relevant to these signs. And also here, this vasodilation is due to certain chemical mediators released at the inflammation, such as nitric oxide. So if we want to screen the anti-inflammatory activity, we can check nitric oxide scavenging activity, because these nitric oxide make the vasodilation and it makes inflammation. So if our plant extract has the ability to reduce these nitric oxide levels, that's mean indirectly it can contribute its anti-inflammatory action. And also here you can see another step, the elevate of local hydrostatic pressure. So the progressive vasodilation in turn may elevate local hydrostatic pressure, and this the transudation of fluid into the extracellular space, and it is responsible for the edema or the swelling which we can see at the inflammation. And here you can see some of the mediators which are involved for the inflammation. We can list many number. Here you can see a few examples, and some are cell-derived mediators and some are plasma-derived mediators. The cell-derived mediators, here you can see the histamine, serotonin, prostaglandin, leukotrienes, and etc. And the plasma protein-derived one, examples are proteases activated during coagulation, kinin system, like that. So all of these are target in the case of screening of anti-inflammatory action of natural products. Next slide, please. And here you can see that arachidonic acids pathway, most of you may be familiarized with this thing. You know, in the arachidonic acids pathway, there are two pathways, the cyclooxygenase pathway and the lipoxygenase pathway. And in the cyclooxygenase pathway also there are COX-1 or the cyclooxygenase 1 and the cyclooxygenase 2. And in this slide you can see in both of these pathways they can make prostaglandin. However, in the COX-1 pathway they make prostaglandin which are important for the homeostatic effects such as the renal homeostatic, then the gastric mucosal protection, platelet function, like that. And when we get this COX-2 pathway, it can make the prostaglandin which can involve for the inflammation, and it can make the pain and the inflammation. So when we get the non-selective COX, in addition to reduction of COX-2, they will reduce this COX-1 also, and then we can see so many side effects. So when we are doing the natural product research related to the anti-inflammatory action, it is better to check the COX-2 inhibition. Sometimes it may not 100% selective. However, compared to the non-selective one, by using the selective one, we can reduce or we can minimize the adverse effects. Okay, so that is some basic background related to the inflammation. Now we will move to the screening of anti-inflammatory action of natural products. In that scenario, this 3R concept is very, very important. The 3R means the replacement, reduction, and refinement. The replacement, this replacement it replaces a method using animals with the scientifically suitable non-animal method. So we can avoid or we can minimize the use of laboratory animals when we are adhere to this replacement method. Then the reduction, any strategy that will result in fewer animals being used in research is also one of the part of this 3R, and it reduces the number of animals to a minimum without compromising the scientific outcomes, because too much usage or the too less usage of lab animal is not good. It is not scientific. So when we are planning a research, we have to think about this reduction, and we have to get appropriate number without wasting animal. Then the refinement, it is a modification of experimental procedures to minimize pain, and it refines the method used in procedure, breeding, transportation, accommodation, and care. Although we use laboratory animal in our research, always we have to think about animal welfare, and we have to minimize the pain that they can get, and we have to think about all their transportation condition, food condition, and housing condition, and everything. Next slide, please. Okay. Then, with adhering to these 3R concepts, when we come to the anti-inflammatory action assays, there are in vitro assays and in vivo assays. There are a number of assays, but with this limited time, I cannot describe all of those. So I selected some of the assays which most of you can do in your laboratory level. So I use simple methods instead of more advanced methods, which are important to screen anti-inflammatory action. And here you can see there are some in vitro methods such as the protein denaturation method and human red blood cell membrane stabilization method. Then there are some in vivo methods like the carrageenan-induced, then the formaldehyde or the adjuvant-induced rat hypoedema or the arthritis models, and also cotton pellet granuloma model. It's this carrageenan-induced one is some of the example for acute inflammation, and this adjuvant-induced arthritis rat model we can use as a chronic inflammatory model, and also this cotton pellet granuloma model also an example for chronic inflammation. That is why at the beginning I mentioned according to our requirement whether we are going to screen acute inflammation or the chronic inflammation, we can select the models that we have to work. Next slide please. Next slide. So here you can see one of the in vitro anti-inflammatory assays. Okay, so if we have in vitro methods, instead of just screening our crude with in vivo, first we can screen that particular activity by using in vitro methods, and if we see the activity, then we, in the progress level we can go to the in vivo model. So in this human red blood cell membrane stabilization methods, the principle behind of this is in the inflammation, the lysosomes can secrete certain chemical mediators which can be contributed to the inflammation. And when we get this lysosomal membrane, it is an analog to the red blood cell membrane. So here we are checking the stability of the red blood cell membrane, and if our results prove that our plant extract or the crude extract has an activity or has ability to stable this red blood cell membrane, then indirectly it says it has ability to stable this lysosomal membrane also. And also when we do this kind of assays, always we have to use a negative control which has the vehicle or the solvent that we are using, and also the positive control which we can add a known drug or the standard for that particular action. And then we can calculate the percentage of stability, and by depend on that we can screen the anti-inflammatory action in vitro mode. Next slide please. And here you can see another method, the nitric oxide scavenging assay by Gris reaction. In earlier I mentioned that nitric oxide is one of the mediator which can make vasodilation, and when there is an inflammation, this nitric oxide can excessively secrete and they can make vasodilation, and they can change the hydrostatic pressure, and finally they can make edema, which is one of the cardinal signs of the inflammation. So in this in vitro method you can see there also we are using a negative control and the positive control and test. So as we are out of the time, I am not going to describe each and every step of this method. These are very common methods, so anyone can read these methods by using research papers. However, I will tell the principles behind of this. So as I mentioned, that is how this nitric oxide scavenging activity can contribute to screen anti-inflammatory action. Next slide please. And here you can see some of the Results we got from 2 of plant material that we used in one of our research. So likewise, once we do these assays for the concentration gradients, then we can plot a graph, concentration against to the nitric oxide scavenging activity, and from there we can read this EC50 value, which gave 50% inhibition. Next slide. And the antioxidant activity, this antioxidant activity is not directly the anti-inflammatory assay, because we know these antioxidant mechanisms are contributed to so many actions. However, when we are discussing about the mechanisms of anti-inflammatory action, this antioxidant activity also there. So I put that one also in here. So all of you know that the free radicals can be defined as any species that contain one or more unpaired electron occupying an atomic or molecular orbit. And within our body there are so many biochemical reactions, and they can make these reactive oxygen species such as the hydroxyl radicals or the peroxy radicals, singlet oxygen likewise. And we know when they have free radicals, they are very unstable. Because of that instability, their reactivity is very high. So they are always trying to damage cells and get that deficit electrons. Next slide, please. But when there are antioxidants, these antioxidants can give that electrons to the free radicals and help them to become stable. Because of that, these our healthy cells can protect from the damage that caused by these free radical molecules. So if our plant extract or the natural product extract has this antioxidant activity, they will contribute to minimize the cell damage. So through that, they can protect cells and prevent the causing of inflammation. And there are so many in vitro antioxidant assays. Here I put only one, but if you read a research paper or a textbook, you can see there are a number of methods. Here I put a very commonly used one, that is DPPH assay. And here you can see the method. Here also there is a negative control and the test and the positive control. So what we are doing, we are making the concentration gradient, and we can check the antioxidant activity for the concentration gradient. And also, when we are working with natural products, most of the time when we are making crude extracts, they have color because they are plant material. So normally they may have green color or a brown or a yellow color. Likewise, they have color. And in these assays, finally we are getting the color absorbance. Because of that, when our plant extracts have color, they can interfere the final absorbance. So how can we avoid that? Normally, when we are preparing a concentration gradient for our test, as you can see in here, in addition to our test gradient, we are using a color blank also. So for the color blanks, we are not putting agent, we are just putting the same amount of our plant extract and the vehicle. Then when we are get the final reading, we can subtract this color blank absorbance from the reading of the test relevant to that particular concentration. And here you can see, then we can plot the concentration gradient, the concentration and the percentage of scavenging activity. And we can plot a graph and we can get EC50 value. So as in all other normal assays, at least we have to do triplicate. And if we want more accurate results, what we can do, get in triplicate, we can repeated the whole assay 3 times, and finally we can get the mean EC50 value from 3 graphs. Next slide, please. Here you can see some of the results that we got, and here you can see a graph that we plot for the Acrontia pedunculata plant extract, and also here you can see a graph for the gallic acid, which we can use as a positive control. And in the DPPH assay, there is a color change. At the beginning, it's purple in color, and because of this antioxidant action, here you can see the reaction, and it's convert into yellow color. Okay. So those are some of the in vitro assays, but as I mentioned, there are so many in vitro assays. So what we can do before we do animal studies, with adhering to the 3R concept, the reduction, the replacement and the refinement, okay? Under the replacement, we can go for the alternatives instead of doing animal studies, so that is in vitro assays. And when we come to the in vivo studies, first of all, we have to choose a correct model, whether it's mice or rats or whatever. Then we have to get the healthy adult animals in both genders, and then we have to house them under the standard condition, and we have to give them a food, the standard food, and the water ad libitum. And also, when we are using animals to the animal experiments, we have to acclimatize them into our animal house condition, because sometimes we are not breed our animals in our animal house. We are purchasing animal from some other place, and we have to transport them to our animal house, and we have to get them. So if we just transport them, and if we use them, they can under stress, and we know we also under stress. There are so many physiological changes, hormonal changes in our body, so animal also like that. So if we just bring them into a new environment, and without giving them to adapt to that new environment, and if we use them, we cannot expect correct results. So always when we bring animals to the animal house, we should give some period them to acclimatize, then only we should use them. And also, when we do animal studies also, we have to use the negative control group and the positive control group and the test group, and we have to randomly select animals for these groups, and we have to equally divide them. Next slide, please. And also, under the reduction, which is a part of 3R concept, when we do animal studies, we have to calculate the correct sample size, and we have to use that amount. Because when we use excessively large sample size, it wastes resource, okay? And also it's unethical. You know, when we do the animal test, always we should get ethical clearance, otherwise we cannot do it. And then when our protocols review at the ethics review committees, they are checking all the things, and they are checking whether we are adhering to this 3R concept. And when there is a very small sample size, the study is not powered enough to be conclusive. So we have to calculate the correct sample size. There are certain methods like the power calculation or the resource equations like that. So by taking one of that, we have to get that. And also there are some guidelines called ARRIVE guidelines, okay? So when we are planning animal studies, always we have to get this checklist, and we have to check whether we are adhering to those things. Next slide, please. And here I put one of the in vivo study which we can use to screen anti-inflammatory action. That is carrageenan-induced rat hind hypoedema method, which is commonly used in vivo method to screen acute inflammation. At there we are using an instrument called plethysmometer. Here you can see that instrument. So what we are doing when we are screening this acute anti-inflammatory action, we are orally administered the testin extract or the testin compound. Then after that we are inducing pore edema by using 0.1 milliliter of 1% carrageenan suspension in 0.9 saline solution, and we subcutaneously inject it and allow to develop edema. And for the then with the time we are measuring the pore edema. First of all, at the base we are getting baseline before we inject, then we are getting readings after inject hourly. So at there what will happen in this picture, you can see the plethysmometer, and you can see there is a container and the electrodes and also the volumetric flask. So to the container and this volumetric flask, we are putting a buffer which is specific for this plethysmometer. Then we are getting our rat, and we get the pore of the rat, and we are dipping the pore of rat in this volumetric flask. So then according to the Archimedes theory, there is a replacement of fluid, and that will detect via this electrodes, and we can get the reading. So then as we have the baseline value also, we can reduce the values, the timely values from the baseline, and then we can get the values. And there you can see the formula that we have to use to calculate the percentage inhibition of edema. So we can reduce the test from the control, and then we can divide by the control, and we can multiply it by 100. Next slide please. And here you can see some of the results that we got for one of the Sri Lankan medicinal plant, and you can see we have to record our results hourly. Normally we are checking about 5-6 hours, and we can calculate the percentage inhibition. And when get the theory behind of this carrageenan method, carrageenan induced rat-hyne pore edema method, actually it's a biphasic event, and in the first phase it's believed that the histamine, the serotonin, the bradykinin, that kind of chemical mediators are involved. And also in the secondary phase, these prostaglandin, the proteases, the lysosomal enzymes, that kind of stuffs are involved in. So once we see the activity in the carrageenan induced rat-hyne pore edema method, then what we can do, we can check in which level our plant material show their maximum percentage inhibition. And here you can see some of the chemical mediators involved for that. So depending on that, then as the next step, we can check some of these markers. Then we can get some idea about this mechanism. Next slide please. And here you can see some of the results and the method of that we have done for the cyclooxygenase 2 activity. So once we see the anti-inflammatory action at the carrageenan rat-hyne pore edema method, then we check this COX-2 activity. Next slide. And here you can see okay. Okay, so when we do natural product research, as I mentioned in I hope the last row hear me. Is that? Can you hear me? Okay. So when we are talking about the natural product research, I mentioned that first we have to check the crude extract. Then by using some of the techniques like the column chromatography, we can do the fractionation of our crude extract, and then we can check the activity for our fraction. And once we see the activity for the fraction, we can select the most active fraction. Then again we can do fractionation for that most active fraction, and again we can check the activity. So likewise, until we end up with our target, that means the isolation of compound, we have to do that. Okay, so in that slide there is a procedure for the compound isolation of one of the Sri Lankan medicinal plant, which is known as Acrontia pedunculata. It's known as Ankenda in Sinhala. I think in India also that plant is available. So when we do that research, we saw that in the phytochemical screening showed that plant is rich with alkaloids. It also contains the flavonoids, polyphenols, tannins, that kind of things, but it has very much we screen the activity of evaluatrine also. In addition to that, we did so many other methods to screen the mechanisms of actions, because one is antihistamine action, because in the inflammation, histamine can be secrete, and this histamine can be make redness, which is one of the cardinal signs of the inflammation. And also I mentioned the pain, also one of the cardinal signs of inflammation. So when we are talking about the anti-inflammatory action, we can check this analgesic action also. So okay, so here you can see the steps that we have followed to isolate the compound. So you can see we check the phytochemical, and this Wagner's test and the Mayer's test, one of the common tests that we can use to screen alkaloids. Then on that TLC, the thin layer chromatography slides, you can see the presence of alkaloids. When we spray drag and drop reagents, these alkaloids will give the orange color with that stain. Next slide, please. And so this is the evaluatrine. So finally, we did the NMR, and by depending on the H1 NMR and the C13 NMR spectrums, we found the structure of evaluatrine. Okay, so that is the story of the natural product research, which are important to screen anti-inflammatory action. And as I mentioned, there are so many other methods also. So what the message I wanted to give you, always when we are doing animal studies, the laboratory animals or the all other animals, they also like us. They also have the same feeling, the same things as us, and as we are feeling the pain, they also feel pain. So always when we do animal studies, we have to think about twice, and we have to check whether the availability of alternatives, and if there are any alternatives, we have to go with our studies by using those alternatives, and when we come to the correct point at the correct time only, we have to use animal studies. So with that message, I would like to especially thank to the organizing committee of International Conference on Recent Innovations in Health and Biological Sciences, which is organized by this PSG College of Arts and Science, and I would like to thank this opportunity to given me, and I'm wishing you a very good evening. Thank you.
Foremost, we apologize to the speaker, delegates and the participants for the technical issue that happened in between. Now we thank you, ma'am, for providing insights on the need for natural anti-inflammatory compounds and elaborating on the cellular responses towards inflammation and its treatment through the 3R strategy. Thank you, ma'am.
Now we request the chairperson, Dr. Changuji, to brief on this technical session.
Is there any question for Dr. Kalpani? It is a good opportunity for understanding inflammation, and I am also very impressed about 3R. Yeah. So I have one question. Is there international rule for 3R or similar thing? Is there international rule or similar thing for 3R? International rule. Yeah, or some books. International.
Sorry, I didn't get that question.
Yeah, or international helpful document. Helpful international document.
International?
Document. Book or book.
Ma'am, I think he's asking about any international guidelines available for this 3 Rs.
Okay. So there are certain guidelines. For example, when we do toxicity testing, there are OECD guidelines as well as WHO guidelines. And these ARRIVE guidelines which I mentioned, when we go through the checklist of the ARRIVE guidelines, it overall cover this 3R concept. So in that guideline, they are asking whether you did the sample size likewise. So these ARRIVE guidelines we can use to cover that.
Any?
Are there any other questions from the student side?
And also any student, you can contact us via email, and we are very much willing to help you for any things that you want to know. So anybody, anytime, you can freely contact me through WhatsApp or email.
Good evening, ma'am. Here. Yes. So as you said, first of all, I would like to thank you for such a good, clear explanation about the anti-inflammation properties of the national products and the 3R concept of animal husbandry and all. And my question is, as you said, we like the inflammation prolonged for a longer time in an uncontrolled manner, we have to use the NSAIDs, non-steroidal anti-inflammatory drugs. So in synthetically produced drugs like ibuprofens are available. Why do we prefer the natural products for this anti-inflammation drugs over the ibuprofen or other NSAIDs which are available in a synthetic manner?
You are asking about the long-term use?
Not about the long-term use. In which way the natural products are efficient or better than the synthetic ones? Chemicals.
Sorry, with the echo I am not clear. I will come there.
Ma'am, I think I'd like to explain what he asked. So he's asking you why we are preferring natural products, plant products in the place of already existing synthetic drugs for anti-inflammatory processes.
Yes.
His question is, what are the advantages of the plant products for anti-inflammatory studies over the synthetic drugs?
You're asking why we are using these herbal things instead of using synthetic ones? Okay, so as I mentioned in earlier, there are some side effects, adverse effects. For example, when we use NSAIDs, the non-inflammatory, the non-steroidal anti-inflammatory drugs, they can make some gastric irritations, and we know it is one of the reasons for the gastritis, these drugs. So when we get these herbal drugs, okay, so this is not only for just the anti-inflammatory compound, we can incorporate these into the formulas also. So normally when we know most of the plant material compared to the synthetic one, they have less side effects. Okay, and sometimes, for example, I did my research with some of the plant that called Psychotria sarmentosa, and it is just a food. We are using that to make some herbal porridge and also to make some salads. So it also has good anti-arthritic effects, and instead of thinking about these effects, when we just get it as a food, we are not seeing any side effects. It's a food. But there are no scientific evidences for that. But through our research, we can give the society the scientific evidences of this plant material, and from our day-to-day life, we can get these things, and it will help us to protect from certain diseases in the preventive level also. And also the main reason of getting these herbal things to get the reduction of side effects. May I answer you, or do you expect it?
75% of the question has been answered. The last one is, what about the efficiency? Like we are switching over to the natural products rather than synthetics. What are the efficiency? The amount we use, the time to take to act?
Yes, sometimes when we check the efficiencies, these synthetic drugs are in front, okay? Because especially when we have worked with the crude plant extracts, the crude mean in addition to our target compounds, there are a lot. So when we get the same concentration, for example, 5 milligram per milliliter, the standard drug and the crude extract, obviously the standard drugs show more activity because it's a pure one, but this is a crude. But when we developed and when we isolate the compound, sometimes we are seeing the more activity, the potent action than to the synthetic one, but sometimes it's not. But compared to the side effects also, so although it has some less activity for that particular action, when thinking about the less side effects compared to that, when we can balance it and we can go with the more dose of that. Okay?
Yes, ma'am. Now it's 100% answered. Thank you.
Okay, thank you.
Good afternoon, ma'am. First of all, thank you for this wonderful presentation. Ma'am. My doubt is about the laboratory animals we use. I actually like the way that we should use alternatives for these laboratory animals by giving them equal emotional and pain values. But ma'am, in our case, it is about inflammation, edema. If inflammation occurs, pain occurs. If inflammation is relieved, pain is relieved. No, ma'am? Clear? So in this case, if we think about the pain that if that animal is getting, we should check if we are inducing an inflammation in a rat's hind leg or something, it experiences pain. When we experiment something and relieve it from the pain, which means we are here we use antioxidants. So we should check, right, ma'am, if the pain is relieved or not. So if we think about the pain that animal gets, we cannot do these researches. So how we will get alternatives for this thing.
So you are asking about this analgesic action, the screening of pain effect? Or what happened when we use the animals and when they have...
You told animals also feel the same pain as we feel. Inflammation means itself, it is pain, right? If something is inflamed, we feel pain.
Yes.
So in that case, if that animal doesn't feel pain, so how we will know the animal?
How you can say that animal doesn't feel pain?
No, as ma'am told, we should find alternatives, right?
Yes.
But we have to test with a living subject that gets pain, and we will check. We will check its vitals from its heartbeat rate and all, if it is experiencing pain or it is relieved from pain. So if we get any alternatives for this concept called pain, then how we can do
okay,
this
so although I say that we have to go to the alternatives, but if it's available, okay, because each and everything we cannot screen in vitro. That is why I mentioned that we have to adhere to the 3R when it's available. So first when we are planning a research, first we have to check whether are there any alternatives. If available, it is not ethical to go animal studies, but if there are no any in vitro methods, like or dislike, we have to go with that animal studies. And also when we are doing the research, okay, sometimes not sometimes, most of the times that they will get pains because of the things that we are inducing. So according to the animal welfare, it is our duty, we have to do the post-treatment. Okay, so when we are doing animal studies, there is a human endpoint, and if we are feeling that animal is suffering more, we have to euthanize them instead of using. And also the post-caring mean, if we do something that give pain, so after our studies we have to give them some analgesics and we have to control their pain until they recover. Okay, so however when we are checking pain, we have to go with these animal studies. Okay, but if there are any other assays that we can check the activity, like the prostaglandin or something inhibition, then if we have that enzymes assays, then first we can check those, and finally at the correct point we can go to the in vivo studies. Okay.
Thank you.
Thank you, Dr. Kapani. Since there are no any question, we are today last session is over. Thank you all.
Thank you for sharing the session, sir. Now, on behalf of the organizing committee, we request Dr. M. Sengutuvan, Head Department of Zoology, to commemorate the speaker with a memento. Thank you, ma'am. No duty is more urgent than that of returning thanks, says James Allen. We gratefully thank the speaker, chairperson and co-chairperson for their support rendered for this technical session.
Now, as we have come to the end of technical sessions for day 1 of this 2-day international conference, we believe that the delegates must have experienced different genres of health and biological sciences of recent trends. On behalf of the organizing committee, we take this opportunity to thank all the resource persons and experts from various domains, institutes and places for sparing their valuable time to fulfill our day with their scientific expertise. We have an announcement for external participants. Those who are in need of attendance certificates and certificate of participation kindly report and verify the details at the registration desk after the cultural event which is about to start right now. Once again, I would like to thank you all, and we assure the delegates that this is not just the end, and we look forward for all your presence tomorrow on day 2, which will be packed with various interesting and innovative sessions. Now, after a whole day of enriching the brain with enormous information, now it's time to sit back and relax. Yes, the visual and audio retreats are waiting for you right now. So have fun, sit back, relax, and let's energize for tomorrow. Thank you all. Please do not disperse. Have fun and relax yourself. Thank you.